Romel, bora. Você vai você vai compartilhar a tela? Tá
Vou,
silenciado.
vou compartilhar. Compartilhando aqui.
Bora dar 5 ou 10 minutos para ver se entram mais pessoas.
Tá bom. É o tempo que eu tô arrumando aqui, então.
Você tem o contato do Bruno? Você poderia entrar em contato com ele? Você está silenciado.
Tenho o contato dele. O que tu quer que eu fale com ele?
Ele é o próximo.
Tá. Peraí.
E depois o Prita.
Que horas são agora nos Estados Unidos?
9h07.
Eu vou te mandar o do Bruno aqui, tá? Só para não ficar telefone sem fio, porque eu vou estar na apresentação. Agora deixa eu lembrar o nome aqui. Bruno Andrade.
Buenos días, bienvenidos sean todos y todas al primer día del Simposio de Ciencias Ómicas de la República Dominicana, como parte del 16 Congreso de Ciencia y Tecnología, organizado por el MESIT y en coordinación con el Instituto de Biotecnología e Industria de la República Dominicana, el IIBI. El día de hoy vamos a tener un día muy interesante con diferentes conferencias. Esta mañana comenzamos con la primera conferencia, que es del profesor doctor Rommel Ramos de la Universidad Federal de Pará, con el título Aplicaciones de bioinformática para el análisis de metagenoma. Un poco del profesor Rommel. Él tiene una licenciatura en análisis de sistema e ingeniería de la computación con especialización en redes de computadores e ingeniería de software de la Universidad Federal de Pará. Posee maestría en genética y biología molecular con énfasis en bioinformática, también de la Universidad Federal de Pará. Y de esta misma universidad posee su doctorado también en el área de genética y biología molecular. El profesor es profesor asociado de la Facultad de Biotecnología y su actual director, y realiza actualmente investigaciones en las áreas de bioinformática, biología computacional y metagenoma. También coordina el laboratorio de ingeniería biológica en el Parque de Ciencias y Tecnología de Pará. Bienvenido, profesor, y el palco es suyo.
Obrigado, Adrian. Gracias. Thanks so much, the organization, for inviting me to speak here. It's a pleasure talking about metagenomic and bioinformatics to you. Thank you, Adrian, for the presentation. Let's go. Well, I will talking about about the metagenomics. But before I work with metagenomics, you need to understand what's the metagenome. The metagenome is a possibility that you have to analyze microbial community in a given environment. You can select any environment to get the sample and analyze the microbial community, microbiota, in this sample. This method is based on it's independent of the cultivation. That's really important because major parts of the microbial organisms are not cultivated. Actually, with NGS and with a lot of data produced by sequence platforms, we have the possibility to generate, to work with the metagenomic data, but we assemble the data and restrict the genomes individually from this sample. What is really important to do some analysis about the evolution in the environment. Well, I need to do an overview here because when you work with metagenomic, you use just the base of genomic analysis to do the same analysis previously. For example, if you are working with genomic, you get the reads from the sequence, you do the quality data evaluation. The step is responsible to remove the low quality bases from your data. You need to do it, depends of the platform that you are using. You remove a lot of reads. After it, you need to choose the methods to assemble. You have a lot of methods. We have the same methods used by different softwares with different strategies. Then you need to evaluate it too to assemble a genome. And after to do the assembly, you need to evaluate your assembly. You need to know how good was your assembly if you represent the sequence that was here, the organism that was here, and you have a lot of tools to do it. Well, if you have a good assembly, you can do the gene prediction. The gene prediction, you identify the coding sequence in your contiguous, in your assembly. After it, you do a lot of analysis. It's really important because I'm talking about genomic, but this step 1, 2, 3, and 4 are common to the metagenomics. The difference of metagenomics is that I am working with many organisms at the same time. It's a really complex to do the analysis, but we use the software, similar softwares, but with different strategies to assemble. Well, for metagenomics, we have two types of analysis of metagenomics. We have one analysis that work to identify the diversity in the environment, and you can use some biomarkers like GSA16S, or you can sequence all information of this environment, of this sample, and this method is called the shotgun sequence. Here, I will show you a simple example how the assembly works. The sequence platforms frequently generate a FASTQ file. The last platforms generate just a FASTQ file. In this case, I am using a FAST file, but I'm showing you that I have a S1, S2, S3 sequence. And the idea of the assembly is compare this sequence, identify regions that you can find identity between the sequence. As you can see in the blue sequences here, I have identity. What's generate a kind of overlap. Then this overlap is a result of the identity of the base. When I grouped the reads based on this identity, I can generate a larger sequence that in this case, we can call the contig. Contig sequence is a result of the group of reads based on identity. Of course, some approaches to assemble genomics or metagenomics using K-MERS approach, but you have the same idea, you have a short sequence or a sequence, and you're looking for identity between the bases to group the sequence and generate a long sequence. Well, to represent this information about the overlap, the identity, we can use in a computational, can use in a graph to represent. The nodes can represent the reads or K-MER, and edge can represent the overlap between the K-MERs or between the reads. The idea is really simple, but when you have a lot of data, you generate really complex graphs, as can you see on the right. The graphs are complex, and the path to transverse the graph can be a challenge. In this case, on the left is really simple to develop an algorithm to transverse the graph, but the other case is not easy. It's the problem. You have a lot of you need a lot of computational research to run this information and to save this information during the process to assembly. Then you have two steps: looking for the overlap between sequence. After this, you have the graph, and you have the last step is transverse the graph. That's really complicated if you are working with the genome. Imagine if you are working with a metagenomic. That probably will share sequences between the organisms that are in the environment, and you have a more complex interaction between these organisms. You have a mobile elements that can difficult your assembly. Then in this case, I am showing just a graph for a genome assembly. Well, there is some things that you need to do before assembling the genome. The first thing that you need to access the sequence coverage. You need to understand that if you have sequence enough to assemble the genome, if you have a genome of 2 million bases, you have a sequence of sequencing results with 4 million bases, you have 2,000 fold of coverage is not enough to assemble. Obviously, because NGS platform, next generation sequence platforms, are based on the redundancy of reads. This redundancy of reads, you can represent the same region a lot of times, It's really important because it's influence the accuracy of your results. Another thing is evaluate the sequence quality. The metric used to do it is the metric called Fred. And the idea in this point is remove all low quality base in the end of the reads or the reads, because if you use this information, you can create spurious paths in the graph if the error occur sometimes. The software to assembly can identify it like a possibility, and you need to remove the low quality base to reduce the chance to occur this problem. The next step is select the k-mer from k-mer value to do the assembly. Of course, you can use the full parameters to test the software, but it's really important you use some software to calculate the best k-mers, the k-mers with more frequency in your data, to select to using the genomic assembly or metagenomic assembly, and avoid to use a lot of computational research to test many k-mers. I don't want to assemble a genome using dozens k-mers. I select just the best k-mers, 5 k-mers, 6 k-mers. It's just because the computational resource is limited. Okay, the last step is select the assembly programs. To select the assembly programs, the selection should be based on the features of your data and the organism that you are working, like the size of this organism, or and the amount of data produced by the sequence platform. It can influence your data too. And the kind of data produced by the platform. We have a lot of platforms, sequence platforms, that can generate reads, short reads, really short reads with 70 base pairs, 100 base pairs, 150 base pairs. You can generate in 300 base pairs. Then you need to understand your data before start the assembly. The assembly needed to be based on a strategy, and to define your strategy, you need to know your data. It's really important. Here I will show an overview about the genome and consider it like a metagenomic tool. In the genome, I have information about the DNA of an organism. I start in a step of DNA amplification. I do a step of site selection. The site selection is really important because it's a step that's occur based on your sequence library that you use. For example, if you are working with a Illumina sequence platforms, you are working with the paired engine library. You have 2 reads that are separated by a distance. When you select the insert size, you select, for example, an insert size of 800. You have this insert size, and the ends, you have the reads. If you have reads of 150 base pairs, you have 150 base pairs in the ends, and the distance between them is the 500 base pairs. Then I can estimate the distance between the pairs. It's really important to use this kind of the library, matching paired ends or matching pair libraries to reduce the problems with the DNA repetition, in tandem repetition that occur in genomes. That's more common in eukaryotic genomes. Okay, after do this step, we do the sequence, and you generate the reads, as I showed you previously. And you take these reads, and looking for overlap between them, generate the graph. This is the step is called layout, and after it, we generate a config. It's really important to understand the these steps, because for metagenomic, you do the same thing, but in this case, you have more than one organism here. To do the assembly of genomics, it's important to show you that you have a lot of options to do it. I can use a velvet, a spade, soap, de novo, all paths, ABs, A5, unicycler, but it's for genomics. For metagenomics, we have a lot of tools to assemble the data. Of course, that are different because we have a different data. We have one sample with many organisms. Then I need to use different strategies to do the assembly. Consider that I have probably redundancy of data due to the sequence. I have redundancy of information due genes that can be shared between the organisms in that environment. Then it's a really problem. For metagenomic assemblers, I can show you some of them: MetaSpades, the IIBI, MegaReads, MetaVelvet, RayMeta. It's just some examples. Of course, we have in recent publications about the review of these assemblers. It's important you verify this kind of review, test some softwares until to start the process to assemble the data. The assembly, I will show you the assembly to compare with the assembly of genomic with assembly of metagenomic. In the assembly, we take the reads and group based on identity and generate a contig. After it, you have a lot of contigs. Then you need to decide the order and orientation of these contigs. I can use different strategies to do it. Frequently, these strategies are based on the information of your reads. I talking about I told you about the basic pairs information. Here we use this information. After generation, the contigs have different contigs here in the contig B, C, A, in scaffold 1. How can you decide the order between B, C, A? Because in this case, I have in a pair read here. My read in red is alignment in the B contig, and in the pair alignment in the C contig. Of course, here I have I can evidence that these contigs are close. It probably can be the sequence. Of course, it's just an example. I can use the libraries with match pair with insert size very long. Then you need to evaluate it. But it's just an example, like the base pairs information, the match pair or pair gender informations can help you to define the order and orientation of your sequences. After the assembly, it's really important you evaluate your assembly for genomic or metagenomic. Frequently, we use the statistical metrics to do it. Then use metrics without reference genome. Just analyze the numbers, the amount of data, the N50, L50, total of contigs I can use to do it. For obtain this information, I like the software Quest. I use this to generate this information. In this case, I like to observe the value of N50 because N50 is a kind of distribution that show you that 50% of your data are into contigs with length greater than or equal to that value. In this example, the N50, the first column is 33,000. 33,000 means that 50% of my data, of the data produced by this assembly, is in two contiguous with length greater than or equal 33,000. That's a great number. It is important because 33,000 bases help me to do the gene prediction. That's the step that I will take some information about the code sequence of my genome or my metagenome. Then it's important to have long sequences during the sequence and after the assembly. If you work with short reads in the first step after the sequence process, you can generate contiguous, large contiguous. But if you are working with reads, long reads produced by PacBio with a 1-2 kilobases, you can generate more contiguous, more longer. Bom, here I will show you another information here. Now I can use a reference to do the evaluation. Look at that, predicted genes. I have the values of unique genes greater than 300 base pairs. Then when you use this information about the reference genome, if you have this information, you can have a lot of results, different results that help you to evaluate the quality of your assembly. The quality in using the statistical information is just about how long are your sequences, the number of your sequences. In this case, with reference, you can evaluate if you are able to identify genes, if you are able to identify mismatches. You can see here. Oh, sorry. Mismatch. I can identify indels. I can identify a number of ends in your assembly. Frequently, this amount of ends is associated to scaffolds that you are working. Because when you create an order and orientation between your contiguous, often you have no information about the information between the contiguous, and you put n. You don't know the base that are there, but you know that there is this base that use n and Quest evaluate it too. For metagenomics, we can do the same thing. I can use the statistical analysis, but I can use the information about the genome of reference. And if you have a reference sequence to use in MetaQuest, you can evaluate misassemblies, events like relocation, translocation, and inversion. And of course, you can do it for genome analysis too, if you have the reference genome. Here, I have a question of binning. Binning is a step for metagenomic analysis. After finishing the genome assembly, I will do the gene prediction, I will do the annotation, I will submit this genome to a public biological database. In this case of the metagenomic, after the assembly, I can analyze the gene prediction, the gene annotation, identify antibiotic resistance genes. I can do many analyses. But if I would like to identify the genomes in this assembly, I need to use this step called binning. The idea of binning is group sequence from the same organism using some features that are able to split the genomes, create difference, show difference between the genomes that were obtained from this sample. Then between this, among these features, can you use sequence coverage of contiguous or your scaffold, nucleotide composition, and tetranucleotide partner or trinucleotide partner, and other comparable features. It's really important to identify the best features that you are working with your data, because with different features that you are analyzing, you create different groups of reads or then different group of binnings. Then you can represent different genomes in your metagenomes. depends on the feature that you are using to do it. How the binning works? In this case, I use just a feature color to separate the data. Imagine that you have in a scaffold 1 and scaffold 2, you have a red or orange contiguous and a blue contiguous. I can just separate this information in 2 groups, the same groups that I am working with metagenomic data. Of course, I use a lot of features. Here, I just use color. Then I can separate the bin 1 and bin 2. The bin 1 is the blue, and the bin 2 is red or orange. Of course, I show you how can I do the binning using scaffold, but I can do the same thing with contiguous. The same idea, it's separate based on feature. What's the main parameter that I can use to separate the groups or the bins? I can use coverage, I can use GC contents, cadence usage, or K-mer frequencies. K-mer IG, 3 tetra pentanucleotides. I can use a lot of information to help me to separate the organisms. Of course, in the metagenomic sample, you can have many organisms that share these informations. Here you have some examples of the tools that you can use to do this analysis. And you need to observe that if you are working with metagenomic, you have 2 steps. The first step is the assembly of metagenomics. The next step is the binning of the metagenomics. The main goal of these tools these tools here, SolidBin, Cocknuts, Coca-Cola, MaxBin2, MetaBats, BMC3C, is define a reliable taxonomic designation for the contiguous of your assembly. Here I will show you an example of the K-mer frequency is a feature that can use to separate the genomes. You analyze the profile of tetranucleotides. In this case, tetranucleotides for K-mer. Let you identify this approach. Let you identify organism profile based on, in this case, tetranucleotide patterns. You can get the sequence here. You can get the sequence here. Sorry. You can get the sequence. You start the analysis using the sequence in the forward, in the reverse. Take 4 bases each time. As you have 4 nitrogen bases ACTG, you have 4 squared grouping possibilities of groups. Here you can see 1 to 256 possibilities of tetranucleotides. And here I will show you the freaks of them. When you finish it, you have a kind of profile of your organism. Interestingly, you can use this information without a complete genome. You can use a draft genome to do the same analysis. You can have a good results. I like this approach using K-mer frequency. Is really efficient. Here I show you the code. If you want to test in your house, in your computer, you can use a library, our library to test it. After the assembly, the binning, after assembly, you can do the gene prediction. But if you want do the binning before gene prediction, you can do it. Here I show you some tools. Glymer MG, Meta GenMark, Prodigal. You can use these tools are really simple to use. Well, here I have important things. In this case, important tips when you are working with an assembly. The contiguous of metagenomics or the genomics needed to be evaluated. I use the quest because contiguous are not perfect. If you remember the graph, the complexity of the graph, you can have spurious paths. You can generate a misassembly during these paths in the graph. Then you need to evaluate. you can evaluate using the statistical metrics, or you can use the quality metrics if you have a reference genome to identify possible misassemblies or some kind of event, non-natural event that or artifact that from your assembly. It's important to always validate the data obtained. If you have no reference, you can use a software like Busco to identify orthologs sequence to validate your assembly. Is interesting. Is a complementary metric to N50. You need to understand the metric before use. It's a support to your choice, and you need to understand what's N50, L50. You need to understand the total of counties, what they mean total of base, what the more important, what's important if you have a set of parameters or metrics that you will use to support your choice. It's important to know this information. Another thing that's important is that long reads are really useful to assemble genomes. It's really important to assemble metagenomes too, but there's many challenges to use it daily for metagenomic analysis. And of course, hybrid approaches using short and long reads is a great strategy. You can use the long reads like a reference to you, and you align the reads against this reference, generate a consensus that can be used to assemble the genome with quality, because long reads has a low quality of the base, but you can use to sequence technology to address this kind of problem. Well, after talking about the genomic, metagenomic, about the methodology, I will show you how to analyze the data. I will present to you tools that can help you. I will present here tools developed by Fabrício Araújo. Dr. Fabrício Araújo developed GoFit. GoFit is a really important tool. You can take your metagenomic analysis, metagenomic contigs or metagenomic scaffolds. You can do a prediction. You get the gene prediction and submit to GoFit. You have some information like this. You have this information. You identify, GoFit will do a blast again NR. We identify another information about your sequence in many databases like Interprop, IFAM, NCBI, EMBO, KEGG. Then I have a lot of information, and I have a kind of word cloud to show the freaks of the gene ontology terms that were found in your data. This tool is really really important to us because it has have been accessed by a lot of people. The last years. Now I will show you some applications of this methodology that I will show you. I will talking about the Dominican Republic, a collaboration that we did with the Dr. Omar Adrian. Here we have a sample collection of the water in the river, some rivers in the city, and to analyze the kind of resistance of antibiotic resistance in the environments. And you can see here an image of the people working, people play in the water with a lot of trash here. Another man working to catch some fish here. Yeah, some plate with the resistance bacteria identified in this work that were conducted by Agent Franklin. The idea here was just identify antibiotic resistance genes, and using a metagenomic data, you get this water sample, go to the lab using a pump to pass the water, and put the DNA in a membrane, and based on this information, agent prepared a work based on some markers of gene resistance in the for this data and generate this publication. It's a good journal this year. It's a Frontiers. It's really important collaboration. Well, of course, it's really different. This project is really different for us because you have just the metagenomic sample, but the analysis was done using the same markers. Of course, the idea is use the metagenomic shotgun sequence here. You have identified our community, but the idea is, can we use the metagenomic shotgun sequence to evaluate the arcs? Yes, we can do it. We did it in Brazil, in Pará, using the two lakes, Bolonha e Água Preta. These lakes are in the environment protection area. That's the area is used as a water supply source for metropolitan region in Belém. And as you can see, you have 1,340 acres in the size. This space serves 75% of the population in the metropolitan region. Then it's really important to us. Of course, you have a disorder in the occupation of this around the lakes. I was talking about it. Here, I show you the lakes. It's really different. You have close to 2 million cubic meters of water, but you have an urbanization without order and deforestation. But in these conditions, with anthropic action, we have an influence of population around the lake. It's a better place to observe spread of antibiotic resistance bacteria. How can I take this information, this sample of metagenomic data, and analyze the resistome, the genes, the set of genes that are some mechanism that have some mechanism of resistance? You can find genes, you can find mechanism, you can find identify mutations associated to antibiotic resistance. Then it's really complicated. You can use bioinformatics if you have a shotgun sequence of this metagenomic. Professor Adriana Folador did it, use these samples to evaluate the resistome of the lake. The idea to do it is just after the sample collection is just bioinformatics analysis. Of course, you can do some wet lab validations, but bioinformatics can give you better support to choose the wet lab validations that you can do. The idea of this manuscript to use just the metagenomic sample. You can go to the environment, in the river, in the soil of the farm. You get this information, the sample. You extract the DNA, of course. You do the sequence. Before the sequence, you need to do the library preparation that are specific for each sequence platform. Do the sequence, obtain the reads, assemble the reads, generate contigs, and you can do the DNA prediction. I'm talking about a metagenomic analysis, but you can see that you can use the same idea to analyze the genome. Of course, after to do the DNA prediction, I can do some analysis. We have a lot of database, a famous database used to analyze antibiotic resistance is a CARD, a comprehensive antibiotic resistance database. Yeah, you can get some information like this. Then I have the gene's name, the best identities, drug class. Then I have a lot of, I have other columns, but I have no space to put here to you. But to show you how simple, after you define your paper pipeline, you can analyze your metagenomic data. You just follow these steps, and you go to use this CARD database. You have the information about the resistome. In this case, I'm looking for genes associated to resistance to antibiotics. just it. Of course, I can work with this data to identify diversity, another bacteria associated to producing bactericin. I have different analysis that I can do. I just show you how can I use to analyze the resistome. Now I will show in a project that we are working now in collaboration with an agent, with a Recon in Brazil. And having this, these are our first steps to genomic surveillance about orgs. While we work in Pará, our stage is really big. We select some points, 8 points, to do the analysis of water, even water, of or rivers. We will do this analysis. We select the samples. You after do the metagenomic collection, you will structure the DNA. You will do the sequence in NGS platforms. You analyze the metagenome. You can compare, for example, resistome. You can associate this resistome, this gene associates to resistance. You can associate it to the region around the rivers to understand what's the activities that are done here. You have farms, you have just a city, close you have industry to identify. The idea is a vision of One Health, a unique vision, that the health of human, health of environment, and health of animal are connected. Then you can do this kind of analysis. You can get many meta-analysis, metadata to do analysis with this molecular information. Here, our stage today, we have this kind of people living under the river, put the trash in the river, having a serious problem about the public health. And to do this analysis, you do the water sample collection, you do metagenomic assembly and binning. Binning just to identify the diversity and compare with markers. After we can do it too, and followed by the gene annotation and the comparative analysis. In this case, our focus will be resistome. Again, the idea is compare this profile of the arcs that you found in the environment on our stage to understand how it's occurring this process of evolution of the problem, and what is affecting the rivers or the lakes to produce this kind of resistance. Of course, you know that resistance is a natural behavior of the organisms that you can find in the environment, but you have worry about how this organisms can influence the human health, and it's important. Our main idea is prospection of biomarkers to identify the impact in these rivers and associate these impacts with probably gene resistance, compare these biomarkers with ecological markers. The idea is create some kind of methodology to identify the problems, identify what caused the problems, what the reasons that the problems continue to occur in the region, and it probably finish each with suggestions how to address these problems, how to reduce the resistance to antimicrobial in the environment. It can affect, including the farms, the activities in the agribusiness. Well, here I have our team, my students, ex-students. Fabricio is a Dr. Fabricio now. From the left to the right, Fabricio, Arian, Will, Fábio, Ana Lídia, Pedro, Adriano, Sebastião, Carlos, Edbruno, o Wagner, o Lucas e a Sandy. Thank you so much, people. If you have questions, I'm here.
Bon, muchísimas gracias, doctor Rommel Ramos. De verdad, un placer poder tenerlo acá. Yo soy parte del equipo del laboratorio del profesor Rommel y del equipo de investigación. Rommel fue mi asesor de tesis, tanto en la maestría como en el doctorado. Y de verdad muy agradecido y muy feliz de poder contar con usted acá. Y con esta, plantear que ya en colaboración con el profesor Rommel, nosotros estamos haciendo varios trabajos acá, tanto para el análisis de calidad de manglares con metagenoma, o sea, estudiar las comunidades que existen en nuestros manglares, y también el trabajo con los ríos Yaque del Norte y Yaque del Sur, donde Rommel es uno de los co-investigadores también. Una persona muy abierta que pueden comunicarse con él. Si tienen alguna pregunta, Graciela.
Sí, buenos días, doctor Rommel.
No se escucha mucho, Graciela. Si puede aumentar.
No sé si...
Ahora sí.
Ok. Muchas gracias. Me encantó su presentación. Ahora, yo tengo algunas... Bueno, voy a limitarme, porque son muchas las preguntas. Quizá después pueda contactarlo por su correo electrónico. Pero en cuanto a las... Oh, I'm sorry. I'm sorry. Regarding the long reads or short reads, is that something that can you control when it comes out of the Illumina output? Did you hear me?
Dígame.
Can you hear me?
Yeah. Romeo.
Okay.
Okay.
Can you, Dr. Ramos, can you hear me?
Yes, yes.
Did you hear my question?
No, please repeat, please.
Okay. Regarding the long or short reads, and I understand your recommendation, but what kind of, how can you determine that what comes out of your Illumina output? I mean, you know,
I understood. I understood. Illumina don't produce the long reads. Illumina produce a kind of reads from 100 basic pairs to 300 basic pairs. I think so, but I have a preference here because I have, if you compare the quality of the data produced by Illumina is better. Of course, if you use long reads, because long reads today is a really interesting term, because long reads to us now is 1 kilobase, 2 kilobases of base is long reads. For me, I work with NGS since 2008. And my reason that time were 21 basic pairs. Now it's 300 basic pairs. It's really long reach to me. Of course, you need to compare the sequence platforms. You need to get the information, but of course, you need to use long reads, just long reads. Today it's very complicated because we have PacBio, you have Oxford Nanopore that produce the reads, but with high error text. It's a real problem. Use just this kind of technology, but I hope that the next years we have the one improvement of the quality of these reads, and you can use just the long reads to assemble the genomes or metagenomics. In metagenomics is really interesting because if you consider the complexity of that environment, you identify if you have one gene shared by many bacteria. If you have a long read, you can identify the region around this gene in discovery the origin of this DNA. We have no duplication.
Okay, thank you.
Thank you.
Muchísimas gracias, Graciela. Si alguien más tiene otra pregunta. Ok. Muchísimas gracias, profesor Romer. Ahora le vamos a dar paso al doctor Bruno Andrades.
Hello.
Oi.
Obrigado,
Hello,
Ed, até
Adrian.
mais.
O professor Bruno Andrade, espere aí. Se me confundió. El profesor Bruno Andrade nos va a presentar la conferencia From Natural Compounds to Bacterial Discovery Using the Computational Tools Targeting Antiviral Prophylaxis and Drugs. Si puede compartir su pantalla, profesor.
Yes, okay. Just a second.
El doctor Bruno Andrade Silva, él es doctor en biotecnología de la Universidad Estatal de Feira de Santana, en Brasil. Tiene experiencia en bioinformática y química computacional con énfasis en biología estructural y descubrimiento de compuestos bioactivos. También tiene experiencia en métodos tradicionales y de inteligencia computacional para el modelado de moléculas de proteínas, peptidos y descubrimientos de fármacos. Actualmente realiza trabajos de cribado de compuestos químicos mediante métodos comparativos, acoplamiento de moléculas o docking y dinámica molecular. Actualmente es profesor de la Universidad Estatal del Sudeste de Bahía en el curso de medicina y coordina el laboratorio de bioinformática y química computacional. Bienvenido, doctor Bruno.
Thank you. Adrian, I need to close the Zoom and open again because I have a problem to share my screen. Okay, just a second.
Okay.
Okay.
Les recordamos a las personas que están en el chat, que están por YouTube, que pueden hacer sus preguntas a través del chat y la estaremos haciendo acá directamente. Actualmente tenemos alrededor de 10 personas en YouTube.
Okay, I'm back. Let's try.
Welcome
Okay.
back.
Thank you. Let's see here. Okay. Can you see the screen? Can you see? Are you seeing my, yes?
Yes.
Okay, thank you. So first of all, I would like to thank you, Adrian, and thank to the organizing committee of this event. And today I'm talking about our research using the bioinformatics to studying the action of drugs and possibility to inhibit the viruses, as well to research about the peptides for prophylaxis against the virus, mainly acting the SARS-CoV-2. Then my title is From Natural Compounds to Peptide Discovery Using Computational Tools Targeting Viral Prophylaxis and Drugs. I'm Bruno Andrade. I'm a full professor of the State University of Southwest Bahia, and I'm coordinator of the bioinformatics lab here. So in this talk, I'll first talk about the viral architecture and the success of the virus and viral biology. And after I'll talk about the focus on SARS-CoV-2 and its targets in our research and what I'm doing here in my lab and with our collaborators. And after I talk about our results, searching for new drugs against the virus and focus with SARS-CoV-2 and using some techniques such as pharmacophore modeling and peptide discovery use artificial intelligence. About the viral architecture, we can see that the virus kind of meta-stable macromolecular assemblies, and this structure is designed for protect its content inside, which is the DNA or RNA. So we have a proteinaceous capsid. So you can see the virus have different forms and different structures and different organizations, as we can see here. So we have a variety of shapes, a variety of forms like spherical, rod-like, elliptic development. So virus is a fantastic organization designed to infect and spread among the living organisms. So the viral structure is designed to contain a protected viral genome and delivered to a specific host cell. So the virus is designed to interact with this specific host. Then these proteins are designed to interact directly with the host. The virus is designed to interact. So as you can see here, the organization of the genome of a virus, we can see that this is a impressive kind of organism. This is not an organism, but it's a kind of molecular machine which can interact with many organisms. So we can have ssDNA, double-strand DNA, SS single-strand RNA, or double-strand RNA. So virus in its genome, it can have many information and deliver this information in many forms. So basically, a virus, we coding for structured proteins such as capsid formation, genome protection, viral shape, or in non-structured proteins such as proteins involving with the viral replication, host interaction, cell entry. So many different proteins we can have coding by virus. So for example, in a capsid assembly, we can find proteins which can close together mount structure for the virus formation. So we call these proteins for scaffold proteins. These scaffold proteins together can give us all the structure of the virus, all the form of the virus, the shape of the virus. So these proteins could be a target for drugs, for example, or to destroy this structure. So this is interactions because interacts because about the success of the virus in the nature. So the virus, the virus kingdom, as you can see, they are infecting different kind of organisms. So we have a virus acting against vertebrates, invertebrates, bacteria, plants, and many other organisms. So we have virus distributed among all living organisms. In the case of the coronavirus, in the SARS-CoV-2, we have virus. This virus is a vertebrate virus and interacting with its RNAs, single-strand RNA. Sorry, let me come back here. Okay. So, and why these viruses are so useful? So virus, we have a biosphere dominated by these viral particles, in the order of 1031. So it's too much particles of this virus in the biosphere. So these viruses are interacting with many kind of organisms. So we can have this virus interacting with bacteria, with fungi, animals.
Professor,
Sorry?
se você está apresentando, não tem trocado o slide.
No?
Não, não está trocando.
Okay, let's come back. Where I stop my slide?
Está no primeiro.
Oh, sorry. This?
And this, yeah,
And change?
não.
Oh, I have a problem. Oh, I'm sorry. It's I don't know why is.
Eu acho que o senhor poderia apresentar desse jeito, se não tem problema.
Okay. But this we won't have the animation. So I try to do another thing. Oh, just a minute.
Ok.
I
Desculpe,
will,
obrigado.
no, thank you. I try to open as PowerPoint because this is a Keynote program. I don't know why it's doing. I will open with PowerPoint. Okay. I'll share again. Can you see my screen?
Oi?
Changing? It's changing now?
Yep, now yeah.
Oh, okay. So let's come back. No problem. No problem. Okay. Back in my talk, I first will talk about the viral architecture and the success of the virus and viral biology, and after I'll talk about the SARS-CoV-2 and its targets, as well as our research about the SARS-CoV-2, and after with antiviral drugs and our study with pharmacophore modeling and peptide discovery applied to the SARS-CoV-2 research. Okay, so what I was talking about is first about the viral architecture. As you can see, in the viral architecture, we have a different kind of viruses, and we have different shapes. And these shapes and this viral architecture is designed to protect its content, its RNA or its DNA, as well as to interact with the host and do cell entry. So we can have many forms of this virus. So we can see that the viruses can have a different kind of genetic material, such as double-strand DNA, single-strand DNA, and single-strand RNA and double-strand RNA. So in this viral genome, basically we have coding for structured proteins, such as capsid formation, genome protection, viral shape, and non-structured proteins such as for host interaction, cell entry, the replicating machinery. So basically, this is what the virus need to replicate and share to many other hosts they are infecting. An interesting thing is about the capsid assembly. So we can find the scaffold proteins, and these scaffold proteins can be targets for drugs and peptides, for example. So these scaffold proteins together will give us the viral form As you can see, let's come back. The form of the virus. So we have this form if gave from this scaffold proteins. Oh, I have a laser here now. Okay, so the success of the viruses is related to its capacity to spread about many things of different kingdoms of in the nature. So we can have the viruses infecting invertebrates, for example, vertebrates, bacteria, and fungi and other organisms. So we can have here this viruses, different kind of genomes, such as SS, RNA, double-strand DNA, single-strand DNA, and many others organizations. So basically, when we apply to the coronavirus, we have this family infect vertebrates, and this kind of virus is single-strand RNA. So as I said, these viruses are dominate the biosphere because they can spread too much. So they can spread among bacteria and fungi and other organisms. And so this creates a massive selective pressures on the cellular organisms. So viruses can adapt to the organisms to infect these organisms and replicate and to do many replication and be stable in this host. So this is what is doing with the new coronavirus. So because we have too many variants, so it's a natural mechanism of selection. So the virus is a machinery that can do this. It's perfectly possible. So additionally, viruses can produce impressive quantity of particles. So as you can see in this example, one infected person spread some particles of the virus, of the coronavirus, to a healthy other person. So just one viral particle interacts with the cell, and then this one particle can replicate inside the cell and spread many other particles. So additionally, this virus can adapt its structure to do protection of its genome, to make recombinant events. This virus can do a large amount of mutation. So this can produce a large number of variants. So the virus can adapt easily to a new host. So basically in the viral biology, we have a hallmark. This hallmark is the presence of the virion. So this virion is a structure designed to enter in the cell and replicate inside the cell. So basically we have this virion enter, such as an endosome, and giving its genetic material to the cell. And this genetic material is integrated, or the genetic material is translated inside the cell. So we have a copy of the virus, this such in the form of protein. So we have the this assembly of this virus, and after we have the viral spread and for other hosts. Okay. Then we have some targets to act when you're talking about development of drugs and prophylaxis. That is basically proteins related to cell entry, as well as proteins related to virus replication. So in this case, we are focusing too much in the spike protein in the case of the new coronavirus. So we select this protein as a target for prophylaxis, for example, or for vaccinology. So this protein basically interacts with the human AC2 receptor. So this is a form of this viral to enter in the cell. And we have some work with the protein involved with the capsid formation. And we have work with the protein related with the replication of this virus. This is the proteases, and there are two very important in this process. So basically, the SARS-CoV-2 genome is a large genome, and it has 29.9 kilobases. This is 82% identical with the first SARS-CoV, and it has 11 protein coding genes and express 12 proteins. So this is basically as this virus infect and spread in the human cell. So basically we have the AC2 receptor and TMPRSS2 receptor interacting with the spike protein and the other proteins with the capsid. So this virus can form endosome. So after the formation of this endosome, this structure is destroyed basically, and we have the RNA of this virus, which is translated by the cell machinery. Then we have the expression of the main proteins and assembly of the virus, and then Golgi apparatus, formation of new vesicles, and then the virus is released to the from the cell. So for this, we have important proteins here, such as the spike protein, E protein. We have M and N protein from the capsid. These proteins are interacting with the different human targets. Okay. So in our work, we are focused on spike, as I said. So in this spike protein, we have different regions. And these regions can be used to drug development and prophylaxis to block the viral entry in the cell. So basically, we can do development of drugs for RBD region, which is responsible to interact with the AC2, human AC2 receptor. We can do drug development for the furin cleavage site, which is responsible for the virus aggressive. So this region, when it's cleavage by the furin protein, so this turn the spike more, as we can say, more infective. So this is the great problem of this SARS-CoV-2. So additionally, we can do work with antibodies and peptides. So we can do using this region for vaccinology and use this sequences to put in a vaccine, recombinant vaccine, as we have now in this technology. So we can do drug discovery and prophylaxis. So basically, what we have with this RBD region and about the furin cleavage site in this new SARS-CoV-2 is we have a mutation in the region of the furin cleavage site. So this insertion here, this RRAR in this SARS-CoV-2, which is different from the first SARS-CoV. So this turned this virus more effective in this case, and so this can be a good region for prophylaxis or drug targets. Okay, So about our research, so just entering our with we are doing here in my lab. So basically what we work is using large scale virtual screening. So we use a big database of molecules to make a virtual screening and docking and molecular dynamics in order to discover some molecules which can act in this spike sites or other proteins from the SARS-CoV-2, such as protease or RNA polymerases. So we use a large amounts of molecules to do it. So basically we use pubicaine and zinc and FDA drugs, approved drugs to do drug reproposition, for example. Then we use these molecules and basically using the computers. So we use these structures for doing doc directly with these structures. One of these proteins that we use is obviously is the spike protein, and then the MPRO. So our idea is to development some kind of the prophylaxis or treatment using drugs and peptides. And our research we using peptides. For this, we are in collaboration with a professor from the Federal University of Minas Gerais. So we are using this software called Propedia. So Propedia is capable to analyze the active site of the protein and propose some peptides which can interact with this site. And then we are using two peptide database, different peptide database. So we use this peptides to dock inside the spike protein, for example. And additionally, we use to peptide modeling to do new peptides which can interact with this active site from spike protein or the SARS-CoV-2 proteins. Okay. So additionally, we are using to glycan database. Glycan are, we know that glycan is a common interact, this common interact with the spike proteins in this structure. So we're trying to find some sugar which can interact in this structure to act as prophylactic molecules. So basically, in this studies, it's designed for viral prophylaxis to development some materials and products which can block the interaction with AC2, the interact with the furin cleavage site. So interaction with the TMP, RRR, SSC2 peptides. And so we can develop, for example, products and clothes and products we can that can be applied to surfaces and foods, for example. So using these peptides or glycans, for example, we can apply these substances here and block the viral contact with the human. So this is our interest with these peptides. Additionally, depends on the kind of peptide, we can use these molecules as prophylaxis, and so it's a possibility, but the main possibility is related to the surface prophylaxis. In the case of the antiviral drugs, our research are focused on a natural resource. So we are using natural products, so a database use that have molecules isolated from plants. And basically, the process of isolation is very expensive, but these molecules are commonly recorded in many databases, which free in the internet. But the process to select and extract these molecules is very expensive. And we know about the biological activity of this molecule is more expensive. So what we can do to analyze biological activity to many natural molecules. So basically we do biological tests. So this is a bench test, but to do it, we need to have many money to do it. And basically the pharmaceutical industry do this and spend billions of dollars to do it. And now a way to do this kind of thing in the way to do a screening when different targets is using a robot in the case of the industrial pharmaceutical industry. So we can test all molecules and have drug candidates. So you can spend too much money. So you can use a computer. So in this computer, we can do a virtual screening and select just the more possible molecules which can interact with these targets. So this is today, this is an easy way to select this molecules for big database with even millions of molecules. And the molecules with higher punctuations can be proposed as drug candidates. And spending not too much money. Okay, so one way to do it is using virtual screening tools. What we are focusing in our study is using pharmacophore modeling. So pharmacophore modeling is a way to describe the molecule, all part of these molecules, and it's with its chemical and structural characteristics, which can interact with the in the protein target. So this pharmacophore modeling can capture the nature and the three-dimensional arrangement of the ligands, which will be reflected in the macromolecular targets that this molecule will interact. So basically, we have in the pharmacophore research, you can see in the right, this is an example of pharmacophore searching. And so we can have here the pharmacophore of no drug, for example. So this pharmacophore is the reference for a database, which you can see in the right. So in this database, the program is searching for similar pharmacophore molecules. So we can propose some of these molecules here and the right as a new drug or as a new possibility to interact with this protein target, which is known to be inhibited by this molecule. So pharmacophore modeling analyzes the 3D structure, hydrophobic areas, hydrogen acceptors and donors, aromatic rings, as well as positive, negative ionized groups. Okay. And so with pharmacophore modeling, we can analyze a large amount of ligands. In this case, we are using ligands from natural resources. So we can generate a pharmacophore modeling and from known drugs, for example, known molecules which are acting in other viruses, for example. So we can have analyze these ligands which were screened by this pharmacophore modeling, if they can act in this protein. So after the process, we will select some hits of the most similar pharmacophore molecules and do experimental validation in the bench. One of our works, we use it for drug development using pharmacophore modeling, is this first paper. So we use pharmacophore with some known drugs, use it for other studies, other proposed as SARS-CoV-2 inhibitors, such as in that case, remdesivir, for example. So we designing this pharmacophore for all of these known drugs, and we made a search against 50,000 natural druggable compounds from zinc. So druggable means that these molecules could be a good drug, so have the characteristics to have absorption and distribution in the human body. And then we made a dock with these best molecules using autodock with the proteases or protease of the SARS-CoV-2. So as a result, we propose about 12 molecules from natural source which can interact with the SARS-CoV-2 protease. In the peptide discovery, we are working with Dr. Raquel Minardi from Federal University of Minas Gerais. As I said, using the Propedia program. And so Propedia analyze the many of PDB structures to propose peptides which can interact in the active site of the protein you can input there. So about 19,000 high resolution PDB complex. And so use this program use artificial intelligence to analyze the sites. So this can propose a peptide sequence to interact with the active site of this protein, and do this peptide modeling and interaction in the active site of the protein. So in order to test it, we got this protein sequence modeling and test with the RBD region, and we tested for furin site 2. And after we did after this peptide docking using the program HPEP-doc, we did molecular dynamics after we have the best docking molecules. As you can see here, this is one of the peptides selected to inhibit the furin cleavage site of the SARS-CoV-2 spike protein. And this is the region, and this is the candidate. So we can perform a molecular dynamics to validate this study. Another thing is about the creation of new peptides use surrogate peptides. This is AI research. So we are working in a program which can got positive peptides that is capable to inhibit the virus, natural peptides, real peptides, and using these real peptides, this program can generate new peptides that can be more strong in this process of inhibition. So first we do use a peptide database to do docking, and this peptide database is a peptide of natural peptides. Is commonly produced by living organisms. And after this selection using the spike protein, we have peptides to propose to be inhibitors of the spike RBD, for example. So we have peptides candidates, and these peptide candidates are our best hits. So this is natural peptides. We select these peptides using energies, and which is more negative is the best in this case. So we use then we use a surrogate assisted model to use this peptide sequence of this positive natural peptides to do new peptides which can block the same region of RBD. So this is a very important way to do new peptides, because this is almost infinite. So this is the program can do it millions of times. So we can have millions and millions of combinations of peptides. So you can test this millions of peptides again and see what is the best hits. So if you have a best hits of these new peptides, you can synthesize these peptides and you can use these peptides in bench and see if these peptides can inhibit the virus. This is not applied only to the SARS-CoV-2. We can use this research applied to other viruses, different viruses. So basically in the surrogate model, we first do the evaluation. We have a database of known peptides. In this case, it's our natural peptides. So we do we select these best peptides. So we make a database of positive and negative peptides, and we give this information to the surrogate model. So the AI algorithm analyze and then training the system to do new peptides. So we evaluate these new peptides, the surrogate peptides, and select the top peptides. So this are doing many times in order to train the system. So in the end, we have some of proposed peptides which you can use and docking again and select the best energies. So we can propose this new peptides surrogates to be used in the bench against the virus. So just to see, as a result, this is not published yet, but we have here 3 peptides designed in this process, which we have a preliminary bench test showed more than 85% of inhibition against the SARS-CoV-2 cell entry. So this is, we have both 2 natural peptides and surrogate peptides. We have positive selection against the SARS-CoV-2 cell entry. So this is a way to using bioinformatics to develop new molecules against viruses or many other organisms. So in this way of peptide discovery, we have another kind of research using chimeric peptides. This is a paper we published last year. And in this paper, we selected the sequence from interaction about the 2 proteins, human ACE2 and SARS-CoV-2 spike. So we analyze this region of interaction. So we select what is the amino acids which are interacting here. Then we design peptides using this sequence and dock inside the region of the SARS-CoV-2 RBD spike. And after, we make chimeric peptides. So we make combinations of this sequence with other peptides which we selected from peptide databases, antiviral peptides database, bacteria databases, peptides database, and others databases. So make combinations with these peptides. So in the end, we have many peptides we can use and propose as inhibitors to the spike RBD. And we make molecular dynamics for some of them. This is just one example. We can see the protein interacting with the peptide. As you can see in red, this is the peptide, and this is interacting with the RBD region. As you can see, this is stable. This is not got far from the protein, staying in the all time of the simulation. So I'm finishing my presentation. I just talk about our lab. We are in a small town in the Bahia, Brazil, and it's called Jequié. This is my university. It's a small university, but we are working hard here, and we are here in this building. So this is let's see. This is the researchers from my lab. So me, Bruno, Dr. Wagner, and Dr. Sergio. This is the students we have now working with these projects with the virals and drugs and prophylactics, and our undergraduate students, Lucas, Zaira, and Felipe. This is my collaboration from Bahia, Dr. Eduardo and Hanner, from the UFMG and Recon, Dr. Vasco, Aristoteles, Dr. Raquel, Debbie, and Sandeep, and from the Virginia Commonwealth University, Federal University of Ouro Preto, Federal University of Uberlândia, Dr. Robson Sabino and Dr. Alexandre Reis. And I would like to thank my supporters here, my university, CNPq, FAPESB, RICOM, CAPES, and the Senai that give us the structure for molecular dynamic simulation. Very important. So thank you very much. And I'm open to questions, and please, if you want, follow us in the social media.
Muchas gracias, doctor Bruno, por su excelente exposición. Si alguien tiene alguna pregunta para el doctor Bruno. Tanto
I
por
have
acá.
one.
Okay.
Víctor Calderón.
Okay, Vito.
Hello, Professor. Thank you
Hello.
for your presentation. It was very enlightening.
Thank
I
you.
would like to know if you have plans, or have you heard of this new tool developed by OpenAI, the AlphaFold tool, which is used to predict protein structure, and if you've been able to see it in action, or maybe plan to use it in the future?
AlphaFold, you said?
Yes.
No, we haven't using this, but this is a possibility to use it. We are testing many other different algorithms and ways to research with it. Basically, what we are doing now is validating all of our research using molecular dynamics. It's just a good way to validate before we have this molecules tested in the bench. But I think it's just a good way to use this research tool. Yes.
All right, thank you.
Okay, you're welcome.
Si alguien más tiene otra pregunta para el doctor Andrade. Okay. Bueno, muchísimas gracias, doctor Andrade, por su presentação. Muchísimas gracias por estar con nosotros. Muito obrigado pela
Obrigado.
palestra. Agradeço muito para o senhor
Obrigado.
a participação. de aqui a pouco vai estar o professor Pritan, só estamos aguardando a entrada dele, com um curso sobre interacciones biológicas. Estamos esperando un momento que entre el profesor Pritan Goss vamos a tener un pequeño curso, un mini curso sobre interacciones biológicas. Va a comenzar a las 11. Introducción a sistemas biológicos estaría comenzando a las 11 de la mañana. Solo regálenos un momento hasta que él se pueda conectar. Muchísimas gracias.
Okay, thank you.
Welcome, Professor Pritan. How are you?
Okay, yeah. Sorry, I was muted. I'm doing great. How are you?
I'm good. All the people are waiting for you here.
Oh, okay. Okay.
We are
I'm sorry.
here and we are in YouTube too.
Sorry, I can't hear you. Yeah, say that again. Yeah.
Okay. You are host now.
Okay, great. So I'm gonna share my screen. Can you see it right now?
Yes.
Okay, great. So let me see how many participants we have. So, okay. So we have not a very high number of participants. So before we start, let's briefly go around the table. And I know Adrian, of course, and I know Romal, but could the rest of you very briefly tell me what is your background? Like, are you an undergraduate student or a graduate student? And in what area are you working in?
Hello.
Yeah.
My name is Graciela Godoy Lutz. I'm a plant pathologist, and I've been recently working mostly in genomics. I mean, I'm beginning.
Okay, okay, great.
Thank you.
Thank you. Yes, Victor, go ahead. I can see you, but I can't hear you. You're muted.
Hello, can you hear me?
Yeah, sure.
Hello, my name is Victor Calderon. I am a student of ADN, and I finished my undergrad in biotechnology. Currently, I'm working privately in a laboratory where we sequence mostly viral genomes right now. Or we're planning to expand, and yeah.
Okay,
basically
sounds good. Okay, thanks. So who wants to go next? I can start calling out the names. Okay, so Julio, you want to go next? You are next on my screen.
Julio and Luis.
Yeah, seems like he is right now muted, but let's move to Luis. Luis, you want to introduce yourself?
Hi, Pritam.
Oh, Roman.
How are you?
I'm good. How are you?
I am professor, your collaborator.
Yeah, I know that. Yeah, that's why I didn't call out your name. I saw your name and I know you.
The pleasure.
Yeah, pleasure here. So you're also associated with the conference, Romel?
Yes.
Okay, that's good. Great. Okay, so I think Julio and Luis are reluctant to talk. So, but then we can get started. So the reason I wanted to get a brief understanding of your backgrounds and all was because I've thrown in quite a few slides over here, not necessarily on genomics. So I wanted to concentrate a little bit more on the systems biology side of things. Now, genomics, systems biology is kind of an abused term. So computational genomics also falls under the umbrella of systems biology to some extent. So what we will concentrate more on in this mini course will be the modeling and simulation side of things. And that's where I will show you some examples of how it is done at a very basic level. However, systems biology is generally a little bit more expansive from that extent. A lot of genomics related work also falls under this umbrella. So without any further ado, so let's get started. So I again have 87 slides. I don't quite think that I will be going over 87 slides in its full entirety during this course. So we can actually pace the discussions depending on your interests and which pieces you are interested in more and things like that. Right? So to start with, I'm keeping it not in full screen mode. Okay, so let me just switch over to full screen mode towards the beginning, because I would like to write a few things on the slides, and staying on the non-full screen mode becomes easier that way. Okay, so let's first get started. What we will do here is very basic understanding of what is systems biology. Then we will move into very standard analysis tools like statistical measures that we use for large genomics data sets primarily, but we'll keep it at a very high level because the goal here is to not get into computational genomics as such. We will spend a little bit more time on some of the advanced analysis tools. And technically, what I mean here is through advanced analysis tools, we're talking more about the modeling and simulation side of things, right? So that's where we will spend most of our time on. And then we will look into a small example, a systems approach to small networks. It's not really small, but through some mathematical abstractions, you can keep the system complexity to a much lower level. Okay, so let's get started. So this is something you guys may have seen before already. So what is systems biology? This is a slide from Dr. Kitano. He was the first to coin the term systems biology. He's from the Systems Biology Institute in Tokyo, Japan. It says that to understand biology at the system level, we must examine the structure and dynamics of cellular and organismal functions. So most of the work go into isolated parts of a cell, but the basic motivation of systems biology is to be looking at it in a more holistic viewpoint, right? So there are currently whole cell simulations that are out there. There is something called VCell, the virtual cell simulation, and that is something you guys can try out. VCell is a very nice interactive web platform where you have the capability of simulating an entire cell. Bacterial cell, they also have the eukaryotic counterparts over there. Of course, the results are not that accurate when it comes to eukaryotic cells and all that, but you can still tailor it towards your needs, right? So properties of systems, going back to the slide here, properties of systems such as robustness becomes very important. Now, whenever you're looking into a large-scale cell simulation or a set of differential equations for that reason, robustness becomes very important because you'll see towards the later half of this mini course that stochasticity plays a very big role, specifically at the concentration level that we work with inside cells, right? So how does the cell keep its robustness in spite of a lot of stochasticity that is going on? So what is stochasticity, by the way? A very basic definition would be, you cannot just assume that A reacts with B to form C all the time, right? So there is a chance, a probability involved of A and B coming together to form C. And that's the very basic level. That's the physics of biology, we call it, right? So every reaction or every regulation of a gene is essentially a stochastic phenomena. There is a probability involved. It's not an absolute deterministic phenomenon. Okay, and that's why robustness, studies of robustness becomes very important. Again, towards the later half of this mini course, we'll talk about stochasticity and what roles it can play and stuff like that. So let's move forward. So there is another slide over here from the SystemX website. They're also talking about what is systems biology, but they cover a few important topics here from what Dr. Kitano mentioned before. So systems biology addresses entire biological systems. Now it's up to you to define what is your biological system. You can just say that my biological system comprises of 3 genes, and I'm just looking at the interaction of these 3 genes, right? Mostly, this is an interdisciplinary field. So you have to have computational people, you have to have mathematicians, you have to have experimentalists, like biologists over here as well. And all of them has to work in unison to be able to do proper systems biology. So experimentalists also call themselves system biologists, computer science people call themselves systems biologists, and so does mathematicians, okay? Now, it involves new types of dynamic computer modeling algorithms, which allows us to simulate entire organisms based on individual cellular components. So dynamic computer modeling, what we really mean by this, the easiest way of doing this is, of course, differential equations, right? And we will see some examples of how a very simple system, we can represent it as a differential equation-based system. How can we solve for it? What does it give us? What these types of modeling tools gives us, right? But differential equations, you'll have to take it with a grain of salt. It is in the continuous domain. So what I mean by continuous domain is that like there are fractional components that will come out. So if you're solving a differential equation, you get values like 1.5, 1.6 for the concentration and all. So it's a continuous domain. When you go to the stochastic part, where you are considering working with probabilities and all, you're looking at discrete values. So number of molecules, it cannot be a fraction. Okay, but if you solve a biological system with a set of differential equations, you will end up with continuous values like fractions and all, which really doesn't make any sense, because how can a concentration be a fraction? I mean, you can look into molar values, the quantities and all of that, and then kind of argue that, yeah, the fractional part still makes sense. But that's the basic difference between differential equation based systems and stochastic simulation systems. So we will cover basics, the basic background of both of these types of systems. Now, strategy of systems biology is dependent on interactive cycles of prediction and experimentation. So you cannot just be a computer science person. So I'm from computer science. So I cannot just go ahead and simulate a system and be done with it, because any predictions that your model makes has to go back to the lab, to the wet lab, for experimentation and validation. Okay. Based on that, a feedback has to come back to the modeling group to say, hey, you are not producing the dynamics properly, so you better change your set of equations accordingly. Okay, so it has to be a feedback loop. This applies to any general modeling simulation work in the systems biology area, and this also applies to the computational genomics area as well, where you're working with a bunch of genes over there, and you're doing differential expression studies and this and that. So based on that, you're making some hypothesis that this gene is important and this gene is connected to this other gene and things like that, right? So whenever you're making those types of hypotheses, you need to go back to the lab and be able to validate those, right? Or come up with alternate hypothesis if it is not validated properly. So the goal of systems biology is the quantitative side of it, no longer qualitative aspects, that gene X regulates gene Y. That's more qualitative, right? When you're looking at quantitative aspects of it, we are going to be able to say that gene X will upregulate gene Y by this much in so much time. Okay, so that's the level of quantitative details that you would want to take. Okay, so go ahead. Yeah, is somebody else co-host here, Adian, too? Because there are some people joining in still.
Me.
Yeah. Okay. Great. Okay. Thanks. Yeah. And during this presentation, feel free to stop me anytime you want, and we can talk more about this. We can keep it quite informal there. Okay, so again, this is reiterating what we just mentioned. So we have a cycle of biology, computation, and technology. Okay, so biology, of course, because you're simulating a biological system. Computing, because you need advanced computational techniques. And we'll talk about some of these pieces when we show you a demonstration for a very simple simulation that we have in place and all. So we'll talk about what types of computational techniques are needed, where is the innovation that comes in. And of course, technology, because you should be able to measure the expression levels of proteins or the mRNA levels of your genes and whatnot, right? So that's where the technology aspect comes in. Okay, so the general workflow here is identify the elements, genes, molecules, cells, ascertain their relationships. Are they co-expressed? Are they interacting between themselves? Integrate the information to obtain a view of the system as a whole. So look at the entire space of your genes, how they are regulating each other. And then large-scale genomic systems have got many challenges. So there are many uncharacterized elements as well that come in over here. So for example, like people have been working with gene regulatory networks a lot, right? What is a gene regulatory network? One gene regulating another gene, up or down regulation and stuff like that. We don't yet know for sure what is the human transcriptional network, right? So what do I mean by human transcriptional network? The genes in the human genome, how they regulate each other, what are the exact edges in a graph? If you think of it as a graph, that is not properly known. We have very well characterized E. coli and east for their regulatory networks, right? But when it comes to the mouse models and to the human level, we don't really know the interactions properly. So that's one outstanding challenge out there, that how can we predict whether one gene is interacting with another gene or not? Okay, and computational analysis over here, of course, requires improved annotation. It needs to reveal the relationships and reduce complexity, right? So that's where the computational pieces are coming in. Of course, small systems, if you have very small systems, the elements are well known. Many relationships are already established before, so this is a priori knowledge. And then that helps with quantitative modeling. So that's why this modeling and simulation aspect of it, many of you may already have background in genomics and you have worked with large-scale gene expression data sets and stuff like that. But when it comes to modeling and simulation, people generally don't keep it to the entire regulatory network level because there are still many aspects that are unknown, right? They generally try to keep it to a synthetic biological circuit, which is a smaller subset of genes that they are working with over there, so that they can produce the dynamics, the robustness, and the different logic of which gene is perturbing which other one much better, okay? So small systems and synthetic biology is where the modeling and simulation world is much more like active. Now, we are talking about gene expression and all of that. So let's just take a very brief look as to what that is. So this is DNA microarray or RNA-seq experiments. DNA microarray was the technology that was used before, and now we have the RNA sequencing technology that is out there. Many of you already have a background for this, so we don't really have to spend much time on this part. But what it does is it gives us a quantification, a measure of the number of mRNAs produced, which basically maps to quantification of the expression level of your genes, right? So how much this gene is getting overexpressed or underexpressed, you can get an idea about that. So it allows for studying genome-wide transcriptional responses, these types of measurements. So RNA-seq data is getting used a lot nowadays. There are many computational tools that are out there. We'll talk about some very basic statistical analysis, but there are several computational tools and packages that you can use. There are web tools that have been developed to work with these types of gene expression values. Okay. So it provides us with a first step towards understanding gene function and regulation on a global scale, right? So moving forward, so what do we do when we get our gene expression value? One of the first steps that we want to take here is to figure out whether they are differentially expressed or not. So what do I mean by differential expression? So you're saying that, okay, this gene in a normal scenario has got a certain expression level, right? So that is what we call control. So this is where I will switch to the basic mode so that I can start scribbling on the sides. Okay, so we're talking about control, which means the expression value in a normal situation. Okay, so it hasn't been perturbed. And then we're talking about expression value under some kind of perturbation. Okay, so every time you're doing an experiment, you perturb the system. So let's say some external stress that you have applied to this system, right? And you're trying to look at its gene expression values based on these external stress. Okay, so those are the gene expression values under e-test, right? Or a knockout. So you have knocked out a gene and you're looking at the expression values based on that, right? So any changes you have made to the system, the gene expression values coming out of those changes is your e-test value. Okay, and then e-control is the normal gene regulation without any perturbations to the system. It is always important to consider both the control and the perturbed system. Only then that will give you a proper understanding of how this works. Okay, so differential expression means that under this perturbation to the system, which genes changed abruptly, okay, in their expression values. Now the change can be upregulation or downregulation, right? So it can be very high, one gene suddenly spiked up and is expressing much more, or it has suddenly spiked down completely from its base level and it is underexpressed, okay? So the upregulation or downregulation, so differential expression can actually categorize both upregulation or downregulation. So the way we do it is we take a log ratio. We don't work with the actual values of the expression, because there is a lot of noise in the actual values of expression, and they basically change a lot. So you might be looking at 1,000-fold difference between the expression value of one gene versus another gene and all, right? So that's why to eradicate that level of noise, we generally work with the log likelihood, the log ratio values, and you take a log of the expression value of one particular gene under the perturbed situation and divided by the expression value of that gene in the control where no perturbations were done. So this, of course, doing normal log algebra comes to log E test minus log E control. Now what this also means is that if E test and E control are kind of at a similar level, that means this gene has not been differentially expressed. So if this is close to 0, if the log ratio value is close to 0, that means this gene is staying in its normal level when it was in control, no perturbations to the system, versus when you have perturbed the system, its expression value has not changed by much. Okay, so that's what we mean when we say that this gene is not differentially expressed. This range right here, E test is less than E control. This means that this is downregulation that is happening, right? That means that after this perturbation, the expression value of this gene has dropped a lot in comparison to the control, the basal case. Okay, so E test less than E control, this means downregulation. E test greater than control, this means upregulation. And then this means no differential expression. I hope you can understand my handwriting, which is not fantastic. So I apologize for that. Okay, Victor is happy with this, so we can go with it. Okay, so upregulation, downregulation versus no differential expression. Now remember, we said that we wanted to work at the log level, the log ratios. But here we are talking about the absolute values, right? So technically, what happens is we put up thresholds. So generally, I can give you an idea that if this R value is greater than 1.2 or so, we say that it is overexpressed. Okay, and if this R value is less than 0.8 or something, we call it down regulation. Log, remember, it's not negative, right? Log is always positive, 0 to something, right? So if it is less than 0.8, we say down regulation. If it is beyond 1.2, we say it's up regulation. This is a rough estimate of what kind of thresholds you would use. And if the R value is somewhere between 0.8 to 1.2, we say that no differential expression has happened. Okay, now this is a very simple idea of differential expression. So now if you're looking at the expression values from RNA-seq experiments or mRNAs and all of genes, you should be able to calculate which genes are differentially expressed. Now there are some advanced statistical methods that gets applied on top of that. So should we just work with a simple threshold value? What if we have done multiple replicates of this? So let's say the same perturbation, but we have done the experiment 5 times. Okay, so now we have got 5 different values of E test. So how do we consider these 5 different values, right? So that's where some advanced statistical testing schemes come in, and there are already available packages that you can use. So if you're well versed with Python, by the way, if you have not picked up Python, this is the time to do that. If you are well versed in Python, you can do everything right now. Okay, so it's very important for the computational genomics area and even for modeling simulation activities and all of that. Okay, so that's one of your homeworks, to go back home and start looking at Python, because there are plenty of tutorials available. Even if you don't know how to code, you can pick up Python in no time. Okay, so it's not that difficult enough. Okay, and there are many differential expression packages that you can now use to be able to check whether one gene has been changed, its expression value has changed a lot due to the perturbation or not. So let's move forward. So generally what happens is we look at an expression matrix like this on your right hand side. Okay, and this expression matrix, what it does is you have the different genes over here and the different conditions. The different conditions means the different perturbations to the system. So this is one way of doing different conditions. Let's say I have different knockouts of genes. Okay, so basically you have knocked out gene 1 and measured the expression values. Then you have knocked out gene 2 and measured the expression values. You have knocked out gene 3 and you have measured the expression values. So these are different conditions, experimental conditions. Now you apply stress to the cell and measure the expression values, right? So there can be multiple ways of simulating your different conditions over there. Okay, so now once you have the genes and the expression values based on the conditions, you get a matrix like this. And then let's say you're just showing the log ratio, which means, of course, we saw the expression for log ratio before. So you're looking at the expression value under the perturbation divided by the expression value under control, and you're taking the logarithm of that, right? So that basically tells you, gives you this type of a matrix, and it's shown as a heat map with the log ratio values. Okay, of course, the more yellow values means overexpressed, the more blue values means underexpressed, and somewhere in between is not differentially expressed, right? So you can visually figure out the level of differential expression from there. In the real world, though, you would see that most genes exhibit little differential expression. Okay, so that's why you need advanced statistical methods to be able to figure out the level of differential expression. It's just not very simply, is it beyond 1.2 or less than 0.8? It's not very simple thresholds that you can use over here. You need some advanced statistical techniques to be able to figure out these types of underexpressed genes and some overexpressed ones, the red one over here maybe, and things like that. Okay. So the next piece here that we generally do at a very basic level with gene expressions is the distribution of their log ratios. So what this graph is showing is you have the log ratio values R in the x-axis, and y-axis how many genes has got this level of log ratio. You will generally see a normal distribution. So it's called basically a normal distribution, the shape of it. What this is showing is most of the genes, about let's say 2,500 genes, has got a log ratio value of 0, which means that they have not been differentially expressed. So e-test, log e-test minus log e-control is equal to 0. So both log values are the same. So that means that they have not been differentially expressed. Okay, so most of your genes will show up on the middle at a log ratio value of 0. And then this spread that you see on the left-hand side and the right-hand side, those are the set of genes that are getting differentially expressed. Okay, so what this plot is showing you is that most of the genes, this is a very narrow plot, which means very little differential expression is happening in this specific scenario. Okay, so moving forward, only a few genes exhibit large differential expression. So the previous plot will only tell you the count of genes which is showing differential expression, right? But how large is the differential expression? That's where this type of a heat map comes in. So now you have all your genes, you have the different conditions, and you have a heat map of their values. So the red values, there are some red values here and some blue values here. So these are the extent of differential expression. Right? So it's not just enough saying that this gene has been differentially expressed. You also want to quantify how much it has been differentially expressed, right? So that is the extent of differential expression that this kind of a heat map is showing. So what do we do with this type of data? So this is data that gets measured very easily, and you do very simple analysis. You, after looking at the mRNA counts of this data, you convert it into log ratio values. You look into whether they are differentially expressed or not. You can figure out the extent of differential expression. This is another example. So when you're looking at the extent of differential expression, look at this plot and compare it with what we saw before. The previous plot had a very narrow peak. This one has a wider spread over here, right? So which means that more genes onto the left and right are showing differential expression. There are fewer genes stacked in the middle of zero differential expression, of zero log ratio value. Okay, so looking at these types of curves, you might be able to say condition 1 versus condition 2, what is happening? So in the previous condition, we saw very few genes were differentially expressed. So that perturbation did not change the system by much. However, this perturbation, deletion of a knockout of a different gene, changed the system a lot more. So more genes are now showing differential expression. Okay, so that's the kind of qualitative understanding that you can gain from these types of analysis. And then when you try to get into more quantitative aspects of it, you generally work with the mean and standard deviation. So you have the expression values, the log ratio values of all your genes. You can compute the mean, the standard deviation very easily. You can compute outliers, right? So there are mathematical ways of figuring out who are the outliers. So this has got at this level log ratio of 6.5. Maybe there are a lot of genes. There are about 200 genes sitting there with a log ratio value of 6. Okay, so maybe something wrong happened in the measurements, because we don't expect so many genes to show such a high upregulation value. Right, so these can be outliers, and there are outlier detection methods, statistical methods that you can use to detect outliers. Okay, so with that, we will cover 2 or 3 very simple statistical methods that are used with these types of data sets. One of the first questions you're trying to answer is condition 1 versus condition 2. How are they different? Okay, so what kind of quantitative knowledge can I gain between condition 1 and condition 2? So you can of course compute the mean and standard deviations of your R values. Each gene has got a specific R value, the log ratio value. I can get the mean and standard deviation based on that. I can get the mean and standard deviation based on this condition. But how do I truly compare these 2 mean and standard deviation? You'll see over here that this is a narrow distribution, and this is a much more wider spread that you see over here, which means more genes were differentially expressed in the second case and fewer genes were differentially expressed in the first case. Okay, so with this, yeah, so there is a question. Victor, go ahead.
Yeah, very quickly. I got confused. The ADE1 and the SSN1, are those conditions or genes?
They are, well, I'm talking about, yeah, you're right, they are genes, but this result was generated by knocking out ADE1, and this was generated by knocking out SSN1.
Right,
so technically they are conditions that way. So that's why we call them different conditions, right? 2 different knockout conditions.
Yes,
however, thanks, yeah. So however, when you're looking at just the mean and standard deviation, it's very difficult to compare these distributions. So what will these values tell us? So you'll see that the mean is quite low in the first case, 10⁻⁻⁵, whereas the mean is much higher in the second case, right? So the value, value of log ratio, the value of R is much higher in this case. But the plots look not so bad because there are many genes still at the 0 level of log ratio changes. Okay, so in a situation like this, it's very difficult to work with simple mean and standard deviation values, right? So that's why we move into different statistical tests to quantify significance. So for example, this scenario shows low variability. So this is condition 2, this is condition 1, and you are looking at the expression values based on that, and you will see that both are quite narrow, right? Which means both have got a small spread, and there is low variability between the 2. In this scenario, you have medium variability, which means that there is still kind of a spread between these 2 scenarios, okay, between these 2 cases. And then finally, you have high variability in this scenario where the spread is much more. So more genes are getting differentially expressed, right? So the amount of variability that is coming out of this scenario is quite high. Okay. So let's move forward. So in order to statistically understand these different distributions, one of the simplest thing we do is student's t-test. Okay, and what does this do? You're basically looking at 2 groups of values. Okay, so this is the value xt, condition xt, and then this is condition xc. So both xt and xc are considered them to be vectors. Okay, so basically I'm trying to say that this is condition t and this is condition c, which is control. Okay, and then I've got 2 vectors. For T, I've got the xi values, the gene expression values. For C, I've also got the gene expression values. Now I'm trying to compare these 2, let's say. Okay, this doesn't have to be control. This can be another condition as well. Right? So when you're trying to compare these two, you use the student's t-test, which essentially tells you the difference between the two distributions. Okay, so how do you do that? The actual value, or the mean of xt minus the mean of xc divided by variance over number of samples of t, variance of c divided by the number of samples of c. Okay, t-statistics, it is the difference between means in units of average error. Okay, so that is essentially the statistical term of it. You can very easily, so current packages will give you the p-value. So if you were to try to measure the difference between two vectors like these, it gives you the p-value, which is a measure of significance, that are they really different or are they quite the same? Okay, so that's what you're trying to figure out over here. Okay, and there are statistical ways of mapping the t-value over to the p-value, right? So different ways of looking at the same thing. Another thing that people do is look at Pearson correlation. So this is basically saying that I have vector, one vector, so I have the gene expression values based on condition 1. I have the gene expression values of condition 2. Are they correlated? Are they behaving very similarly? Okay, correlated does not mean a one-to-one match between them. Correlated means that are they behaving similarly? Okay, in order to do Pearson's correlation, you actually look into some more complicated expression right here. It looks much better if you were to use z-scores. So you can just look up how to calculate z-scores. Given a vector, you can calculate z-score based on the xi value, its mean, and the standard deviation. It's very easy to map it to z-scores and all. And this is the actual expression for computing the Pearson correlation. So what is Pearson correlation really trying to do? What is the intuition behind this? So let's say I have gene 1 and gene 2. Okay, so you'll see that the Pearson correlation in this case is almost close to 1. Why so? Because if you look at the, so these are conditions, condition 1, condition 2, 3, 4, 5 different conditions. In these 5 conditions, gene 1 and gene 2 are behaving very similarly. Okay, so the yellow is upregulation, high values of its log ratio. The blue is the lower values of its log ratio. So under each of these conditions, both gene 1 and gene 2 are behaving very similarly. Okay, so then if you plug these expression values into the Pearson correlation formula that we saw in the previous slide, you will get something close to 1, which means that they are very well correlated, right? Across different conditions, they are behaving very similarly. That's what we mean. Okay, look at this scenario. Gene 1 and gene 2 are not at all correlated. So it has got a Pearson correlation value of 0. What do we mean? Why is it 0? Because you see that it is getting upregulated here and downregulated here. Okay, it's the opposite. But does it mean that, is it exactly the opposite? No, it is getting downregulated here and close to staying constant here. So it does not mean that it will just behave the opposite, right, across these conditions and all. It is getting upregulated here and again upregulated here. Not by exactly the same amount, but still upregulation. So it is erratic behavior of gene 1 and gene 2, right, across the 5 conditions. So they are not correlated. So that's why we say that the correlation value is 0 in this scenario. The third case, we have a negative correlation between gene 1 and gene 2, which means that gene 1 and gene 2 behaves exactly oppositely, right, for the different conditions. So it is getting upregulated here, getting downregulated here, kind of downregulation or close to normal, and this is also close to normal kind of downregulation. Up and down here, different ones, down and up here, so on and so forth. So it is just behaving oppositely in most cases, right? So that's why they are very well correlated, which means that looking at gene 1, can I predict what will happen to gene 2? That's what we are trying to say by correlation. And we can say that, okay, yeah, we can. In this scenario, we can, because if gene 1, if I see gene 1 upregulated, I can predict gene 2 will be downregulated, right? So they are negatively correlated here. Here, I cannot say anything because they are not correlated. So just looking at gene 1, I cannot predict what will happen to gene 2 in a new condition, okay? Here, they are positively correlated, which means that looking at gene 1, I can predict still what will happen to gene 2. It will behave very similarly as gene 1 in a new condition, okay? So these are the benefits of being able to compute correlations. in these types of scenarios. However, you have to take it with a grain of salt. Correlations are not a magic number. And as you can see over here, that a correlation value, so this is just a trend line. And if you look at the correlation here, the correlation, the R square of these fits are 0.8 in all these cases, in all of these different pictures, right? So because the R square is still 0.8 in all these cases, you would say that all of these data points are correlated very similarly with this trend line. But that is not truly the case. I would go with something like this because there is one outlier, which is fine. I can work with outliers, but the other ones are falling in my trend line much better. But over here, they're not following the trend line at all. Over here, they're still following to some extent, But this is the worst. This one is the worst, right? It's not following the trend line at all, right? So the point is, you cannot still go with an absolute value that comes out based on the Pearson correlation, right? So you can at best say that it is positively correlated or negatively correlated or not correlated at all. This is a qualitative inference. This is not a quantitative inference. So do not take it as a quantitative inference, these types of correlation studies, okay? For quantitative inference, that's where we will jump into the kinetic models, where all the fun will start. So computational genomics, let's just finish up the first part of it. One of the other things that you do with these types of gene expression values is you cluster them. Why do we cluster them? We are basically trying to figure out which genes are behaving similarly, right? So you're trying to figure out which genes are behaving similarly. That's where you use the correlation, let's say the Pearson correlation values and all. Based on the Pearson correlation values, you saw that, okay, gene B and gene C are very similar. Okay, so you'll see, looking at their expression values across the different conditions here, it's not a perfect match, but most of the time you see right here in this block, they're very similar. In this block, they're very similar. So when you're computing the Pearson correlation, it's across all these conditions and all, right? So if in most of the cases we are behaving very similarly, it will end up having a high correlation value. Okay, so what clustering does, hierarchical clustering does, is it basically puts together the genes which are very strongly correlated, very high value of Pearson correlation. So I say that, okay, these two genes are very highly correlated. Okay, after this is done, you recompute the correlations of this bunch to the other genes. Okay, and then redo the process. That, okay, this is my first level. In my hierarchical level, these are the closest correlated genes. Then I go to the next closest correlated genes. Then I go to the third level correlated genes and all that. So that's how people do these types of hierarchical clusterings in these types of systems. This is a more real world scenario where you can see this is how the clusters are looking like. So basically, which genes are basically packed together. They are kind of correlated. They are working like together in most of the scenarios and all. Okay. Okay, so there is another clustering which is quite common. Some people who have worked with these things would know about k-means clustering. We just saw hierarchical clustering. K-means is another way of clustering the genes. It is solving the same problem. It has got the same purpose of figuring out which genes are working together. They are kind of related to each other, right? K-means clustering, the problem here is you have to define a value of k, how many clusters you want. And then once you have said that I want 3 clusters, these are my 3 centroids of the clusters, I'm just looking at the points, seeing which cluster should it belong to. This is a new point that has come in, a new expression value. This is closest to this centroid, right? So I put it in this cluster. Whereas if this gene has come in, it is closer to this point. So I put it in that cluster, like that, right? So that's how the k-means clustering works. So we are talking about more the methodology of these clustering algorithms here and all that. Again, why do we need this? Because we are trying to pack the genes together which are very much correlated, which are working kind of together. So that's why they are correlated to some extent, right? So that's another biological question that people try to answer based on genomics data sets. Okay, so we rushed through the genomics pipeline because we wanted to get into the kinetic models, right? Looking at most of you backgrounds, I feel that most of the genomics pieces are kind of things that you would know already, and there are a lot of pipelines and web tools that are available that you can now use to do these types of genomics studies. Okay, so that's why we wanted to spend more time on the actual modeling simulation aspect of things. That is where I think many of the systems biology or bioinformatics programs don't go into kinetic models as much, and that's why we wanted to spend more time on this aspect. So let's move forward. So kinetic models, let me just brief intro here. You're going to see a lot of equations. Don't feel bad about it, because at the end of the day, it is all physics. Okay, so we are looking at biological systems, but at the end of the day, we are looking at how they are behaving in a mathematical standpoint, right? And then once you've written down your equations, which is not too difficult, once you get a hang of it, you understand how they work and all. Once you've written down the equations, you can now simulate this on a computer. And simulation using kinetic models, this gives you an exact quantification of what's happening in the system in terms of the quantities or the concentration of the different proteins, the mRNA levels of the different genes. You can exactly say with time how this is changing. Okay, so that's the level of details that you can get from these types of models. So the most popular model that is out there is called the mass action model. So consider a very simple reaction, A plus B forming C. Okay, this is just an abstraction, by the way. It doesn't have to be a chemical reaction as such. I can say that A plus B are two different proteins which is forming a complex C. Okay, I can say that A plus B are two different genes which is regulating a third gene, C. Okay, so it's just the abstraction. Okay, so these types of modeling and simulation are getting used in biochemical reaction networks, getting used for gene regulatory networks, you name it, it's getting used everywhere. Okay, so A plus B forming C, what exactly is happening is a molecule of A collides with a molecule of B. That's the physics of it, right? So when you're trying to model this, you got to understand the physics behind this process. So molecule A is colliding with molecule B to form a molecule of C. Okay, generally we look at large macromolecules, which makes our mathematics much easier, and that brings up the law of mass action. Now I can write this as a differential equation. Okay, so that's the domain of mathematical biology. So whenever I see a reaction like this, so this is called the rate constant. This is a constant value that you need to say that, okay, how quickly is this reaction progressing, right? So that's what the rate constant is. That is not the rate. Rate is the number of reactions per second. Okay, this gives you an idea of the rate. There is a difference between a rate constant and a rate. Okay, what does it mean? That means that if I have more molecules of A and B, I will obviously have more reactions happening, right? So that's why I need the concentration of A and the concentration of B in this equation. If I have more molecules of A and B, I will end up having more reactions. And this K is a constant factor. It's a constant which categorizes this reaction. It says that, okay, this is how this reaction behaves. Okay, it basically puts up a bound on the number of reactions that can still happen. If I throw in infinite number of A and B molecules, K still puts up a bound on the number of reactions that can happen. Okay, so that's the understanding of it. So now let's look at a little bit more complicated system. Now, most of our biological networks and biological systems are reversible reactions. So which means A plus B forms C, but at the same time, C can break down into A plus B. So there is a reversible reaction happening. Now, the positive direction rate is K plus to form new molecules of C. The backward rate is K minus, where C is breaking down into A and B molecules, right? So in most biochemical reactions, reverse reactions also take place. And now my differential equation becomes a little bit more complicated. So when I say dA dt, I can write differential equation for all of the three quantities, all of the three species. We call each of these species. A is a species, A is a species, B is a species, and C is a third species. So whenever you have multiple species like this in your system, you can write differential equations for each one of them separately. So this is my differential equation for A, only the species A. How is it working? New A is getting formed with this expression, K minus times C. So think of it. Why is this expression positive? Because we are saying that it adds to the value of A. What is dA dt? It is the change in A, right, with time. How much is A changing in delta t, in time? That's what we are trying to figure out with this differential equation. So this is a positive term. This means that this is adding more A molecules. How is it doing that? Through the backward reaction. So C is breaking down into A and B. So through the backward reaction, more the number of C, more is the concentration of C, more A molecules will be formed, right? And with a rate constant of K minus, which is my backward rate constant, right? So that's the amount of A that is getting formed. Now the second part is how much A is actually getting away out of the system, right? So this is the forward reaction. So this is basically taking A molecules out of the system, right? And what is the rate of my forward reaction? We already saw this in the previous slide. K plus, which is the rate constant of the forward reaction, multiplied by concentrations of A and concentration of B. Okay, so that's my forward reaction rate. Make sense? So that's how you construct a differential equation, right? So given a reaction like this, how do I construct a differential equation? I write down dA dt, which is the change in A with time. I figure out how many new A molecules are getting formed. This is the expression for that. I figure out what is the rate of A molecules taken out of the system. This is the expression for that. Okay, the two of them together tells me how will the A molecules change with time in this system. Okay, at equilibrium. So now, so I can now write the differential expressions for the other guys as well. How about dB dt? So for dB dt, again, it's exactly the same. K minus multiplied by C minus of K plus times A times B. Exactly the same as dA dt, because it is also getting governed by the same forward and backward reactions. It is also getting formed by C breaking down, and we are also losing B molecules because of the forward reaction right here. So dA dt and dB dt are exactly the same. Now I can write down the third differential equation, dC dt. So what is dC dt? dC dt, it is getting formed by the forward reaction. So I will say K plus times A times B. So this is the positive term, because this is how C is getting formed. And how is C disappearing from the system? Minus K minus times C. So it's just the opposite. So the plus has changed to a minus, the minus has changed to a plus. From the expression that you see right here, dC dt is just the opposite. So I have the positive term as K plus AB and the negative term as K minus C. Makes sense? Because C is just behaving the opposite over here. So technically, whenever you have a biological system, so this was a single reaction. Now I can have a biological system with 1,000 different reactions like this. Okay, and now your goal is to write down these differential equations based on these simple expressions. Okay, once you have written differential expressions, you can use MATLAB, and there are many other simulators to simulate the system. You throw in an initial concentration to the system, and it will tell you with time how is the concentration changing for each case. Okay, so that is the basics of the simulation. But most of the time, what people do is they try to do equilibrium analysis. So before we go into equilibrium analysis, one of the problems that happens is you can measure A, you can measure B, you can measure C in most scenarios, right? What you don't know, the unknowns, are these two quantities, the K plus and K minus. So that's why the first step of doing these types of systems biology simulations is to be able to measure K plus and K minus. What is the rate constant of a reaction, the forward rate constant, the backward rate constant? So that's the most important part. And that's where you need experiments. So experiments being able to design the experiments properly, it can tell you what will be the values of your K plus and K minus. So in this sequence of slides, we will see how to actually get estimates of K plus and K minus. That's where we generally do equilibrium analysis. Okay, and you'll see the importance of equilibrium analysis as we keep moving forward. So at equilibrium, so what is equilibrium again? Theoretically, equilibrium means that after a certain point of time, the concentrations are not changing anymore. Okay, so dA dt is equal to 0, dB dt is equal to 0, dC dt is equal to 0. Okay, if dA dt is equal to 0, so let's say this first expression right here, if we make it equal to 0. Okay, that is what is happening at equilibrium, which means that the concentration of A is staying constant. It is saturated. It's not changing anymore. It's not going up or down anymore. So if we make this expression is equal to 0, I can simply do the algebra to say C is equal to K plus by K minus. See the K plus here, this K minus comes to the denominator multiplied by A times B. And we are saying EQ, we are saying that these are the equilibrium concentrations, right? So when we say that this is equal to 0, this condition only happens when? At equilibrium. It is not happening throughout the time range of the simulation, right? This is true, this is equal to 0. This is true only when we reach the equilibrium, not before. That's why the concentration values that you're going to use are the equilibrium concentrations of these two cases, right? So EQ, BEQ, and CEQ are the equilibrium concentrations of these three species. That is not the initial concentration of the three species or the way the concentrations changed in between. So I'm not looking at those. I'm only looking at equilibrium when the concentrations have flattened out. They are staying to a constant level. Okay. So with this, let's consider our initial concentration. So my initial concentration of A is A0, let's say. Okay. So this is my initial concentration of A. Okay. Why is it equal to C plus A? So what is the difference between the two? What is C? We are basically saying equilibrium concentration of C, and we are saying equilibrium concentration of A. So this expression is saying that the initial concentration of A is A0. A0 is equal to the equilibrium concentration of C plus the equilibrium concentration of A. Why is it so? At equilibrium, A is not changing anymore, right? So there is some A that is still left. So let's say A, if I were to plot A, so it started from here, and at equilibrium, it is right here. It did not go down to 0. That did not happen. So it is staying flat here. So this is my equilibrium concentration of A. Okay, it's not changing anymore. What happened to C? C started from 0 and it has saturated right here. That's my equilibrium concentration of C. So what is this expression saying? It is saying that well, I shouldn't have started from here. I should start A from here. So just use the eraser, oops. Take this piece out. For some reason it doesn't allow me to. Anyways, so this is A and this is C. Okay. Initial concentration of A, which is A0, is equal to final concentration of A at equilibrium plus the final concentration of C. Why so? Because if you look at this reaction, every A that I lost created a new C molecule, right? So considering that, this is the amount of A I'm left with. So this is not producing any more Cs. This is staying constant. And the Cs that have been produced correspond to the As that were lost. Okay, the two of them together gives me A0, which is my initial concentration of A. Okay, so once you consider this expression right here, so now I can write C, which is the concentration of C at equilibrium, is equal to, so I'm just plugging in this value of A0 is equal to C plus A. So let me try to write this. So I have got too many scribbles right here. So I'm going to take this off. Okay, so now how do we get this final expression of C? What is the concentration of C at equilibrium? We want to keep it to one quantity only. Okay, so basically over here, what is the problem with this expression? I have to measure both A and B, right? So we want to keep our experimental requirements smaller. So here in this expression, I just want to measure B, right? Just based on measuring B, I should be able to predict what is C. Okay, what is the equilibrium concentration of C? So that's what we are trying to do over here. So considering this and this, I can now write C. So I'm dropping this EQ and the brackets and all, so just to keep it simple. So C is equal to K plus over K minus. So let's say that capital K is equal to K minus over K plus. So which means A times B divided by K, capital K, right here. Okay, and then what is the value of A? We know that from this expression right here. So we write A is equal to A0 minus C minus B over K. And then if you just simply do the algebra from here, because you want to take this C over to the left-hand side as well, right? Because you're solving for C. If you simply do the algebra by taking the C to the left-hand side, you will get this expression right here. Okay, so that's the benefit of equilibrium analysis. So this becomes a very easy experiment now from a biological standpoint to do, that I will only be looking at the system at equilibrium, and I just need to measure the value of B at equilibrium. So just looking at the system at equilibrium, the concentration of B, I should be able to predict the concentration of C. Okay, and also I need to know the initial concentration of A0. So based on that, I should be able to predict this system, like how it is changing with time. But remember, our question was how to figure out these rate constants, and that's where these types of reductions become important. So let's move forward to check out how people try to measure the rate constants in these types of scenarios. So we are looking at a very simple system. We are looking at the enzyme kinetics system. It's very well characterized. People know about it already. So what's happening here is a substrate plus an enzyme, S plus E, forms a complex C, which is the intermediate, and then C breaks down into the product P and leaves the enzyme out. Right? So that's your normal enzyme kinetics equation. We have already learned how to write my differential equations for each species. So that's what we are writing here. We need to write a differential equation for S, for E, for C, and for P. Right? For each one of them, we can now write these differential equations very easily, following how many molecules are getting formed, how many molecules are disappearing, right, at each step. So based on that, you can write all of these things. And then in this last stage, you will see from enzyme kinetics, we generally don't use the reverse reaction, because like once the product and the enzyme is created, they don't quite combine. We take the product out of the system, because that's the final product that you're looking at. So you don't keep the product in the system for long enough, so it's flushed out of the system. That's why this reverse reaction is generally not considered for enzyme kinetics types of systems. Okay, so once you have written down the differential expression over here, now you can plot the Vmax. Vmax is the number of reactions per second. Okay, number of reactions per second, we are basically trying to say the value of P, which is my product. Okay, how much of the product is getting formed per second. Okay, so that is something you can plot based on the enzyme kinetics reaction. At Vmax over 2, so generally, when you're looking at systems biology, you don't look at the maximum value that I can reach for the product. For enzyme kinetics, you're looking at half the value. And you're based on this half the value, you try to figure out what was the rate constant in the system. And there is a reason why you want to look at half the value. This is the realm of Michaelis-Menten reactions and Michaelis-Menten constant. So you may have seen, if you're working with systems biology and these types of simulations, that people say that, okay, we assume that this reaction follows Michaelis-Menten kinetics, and they write an expression based on that. Okay, so why is that simplified expression? How is that simplified expression coming along, right? So generally, Vmax is the maximum rate of this enzyme-mediated reaction, and by increasing the and you can actually keep pushing up this rate of reaction if you keep increasing the substrate concentration, right? So remember, if you go back to the basic expression right here, if I keep increasing the value of S, I should get more P in the system, right? So Vmax is saying what is the max value of S that I need to create the maximum amount of P in the system, right? So that's my maximum rate of reactions. However, in reality, what is happening is the speed V means the number of reactions per second. You already know that. But what is actually happening is the system never reaches this Vmax. Okay, this enzyme kinetics system will never reach the Vmax because there are other rate limiting factors in the system. Because the complex is getting formed, there are some rate constants associated with the complex being formed. The number of enzymes plays a role into the system, in these equations as well. So if you are trying to say mathematically, you're looking at the expression, looking at the simple reaction, you may think that, okay, I should be able to achieve such a high value for my reactions, okay, for my rate of reactions. But practically speaking, most of the time you don't achieve that kind of value. You may achieve only half of it. So Vmax over 2 is what you may, you generally do achieve Vmax over 2, and that can be measured, right? So that's why you try to characterize the system based on the value of Vmax over 2. The Km value is called the Michaelis-Menten constant, and we'll see how to compute the value of Km, which is the rate constant at Vmax over 2. Okay. So now let's check out again equilibrium approximations. I promised a little bit of equations. So now we are getting into a few simple equations, but we will just work it out. So it's not so bad. ds over dt is equal to 0. What happens in equilibrium? As we mentioned before, we are considering that each of the quantities are not changing anymore, right? So if s is not changing anymore, we are saying ds over dt is equal to 0. So if you go back to the expression for ds over dt, this is equal to 0 means these two expressions are equal, right? So k minus 1 times c is equal to k1 times se. Okay, so when you write that, so this is what you write. Now you're looking at the initial concentrations. So I have an initial concentration of enzyme E0 is equal to E plus C. It follows the same concept that we talked about a little while back, that when we were looking at A plus B equal to C kind of a reaction, that if my initial concentration of the enzyme is E0, then I can actually say that E0 is equal to the equilibrium value of enzyme, how much enzyme is left at the equilibrium, plus how much of the complex is left at the equilibrium. Okay, once you have plugged that in, you put up the C, you do a simple algebra over here to come up with your value of V. So dP dt is equal to K2C. Go back over here, dP dt is equal to K2C. Okay, and that's my rate of reaction. Remember, it's a rate of reaction. V is a rate of reaction because this is dP dt. It's not a constant value, right? So this is how much P is changing with time. So P is still getting formed. That's my rate of reaction. That is equal to K2C. Once you have solved for C from here, I can plug those in and I get something like this. We were talking about Vmax. Vmax is equal to K2 times E0, which means that what is the maximum this rate of reaction I can create? It depends on the number of enzymes I have thrown into the system, right? So if E0 is the number of enzymes that I've thrown into the system and K2 is the rate at which the enzymes are producing the product, then K2 times E0 is the maximum product formation that I can get. However, as I mentioned before, we generally don't reach this Vmax most of the time. We only look at half of this Vmax. So moving forward, how do you do the rate constant determination? This is the expression that we saw in the previous slide. You just do simple algebra, you get something like this. 1 over V plus 1 over Vmax plus this second quantity, simple algebra, so we don't really have to work it out. This can be written as a simple line equation. Y is equal to A plus Bx, right? So once you have a simple line equation like this, you plot it. So you basically run this reaction multiple times and you are plotting the values of Y and X over there, right? And based on that, you're coming up with the constants A and B. So this is just a fitting that you're doing this way, right? Once you have come up with the values of A and B, the value of B allows you to find Km, right? Because that's one of the constants which is inside the value of B. Make sense? So at a very high level, when you're trying to compute your rate constants, technically for the same reaction, you're doing multiple experiments with different values of A and B in the system. Let's say for the enzyme kinetics, for different values of enzymes and substrates in the system. Those are my initial conditions, enzymes and substrates. For different values of that, I'm running the system, right? I'm measuring the product that is getting formed, and then I'm putting it back into this Burke plot, Lineweaver Burke plot, which is this y is equal to a plus bx. I'm just doing a curve fitting based on that. And after having done 5 or 6 different experiments, I should be able to reliably compute the rate constant of the system. Okay. Once you've got the rate constant of the system, this way now you can predict how this Michaelis-Menten system, the enzyme kinetics system, is going to behave for any different concentration values of my substrate or my enzyme and so on and so forth. Now I can keep changing those values. Once I've trained my model this way, I can keep changing the values of enzymes and substrates and all of that, and I will get different behavior of the system. What is the concentration of the complex? What is the concentration of the product at any time point and all of that? Okay, so moving forward, so with enzyme inhibition, this has been one of the most well-studied systems, and we are spending more time on this one just to give you a feel of the kind of complexities that can be brought in, and these can all be solved using differential equations. Okay, so we are just trying to show you some fields of how can differential equations be used for many different types of complexities for a very simple enzyme kinetics system, right? So once you understand this, it can be blown up to much more complicated systems and all, moving forward. Okay, so a substance that inhibits the catalytic action of the enzyme, that's what inhibitors are, right? So there are 2 types of inhibition mechanisms that generally happen. There is something called active sites-based inhibition and allosteric or regulatory sites-based inhibition. Okay, so that's where we are getting into the biology side of it a little bit. Active sites, what it does is, there are 1 or more active sites on the large enzyme molecule. So let's say this is my enzyme E, and this is the binding site for S. The substrate has to come and attach here. Okay, so what's happening here in this scenario is the substrate does not get to attach. The inhibitor molecule here comes and attaches to this site. So if a different inhibitor molecule attaches to the site, the substrate can no longer make a complex with the enzyme, and that means that I'm not going to be producing the product much, right? So pictorially, what's happening? Okay. So essentially, what's happening is S plus E forms C1, from there forms E plus P, right? So that's my complex in the original sense. But instead of the substrate, if I had an inhibitor molecule coming in here, I form a different complex, C2. This different complex is stuck right here. It does not produce the product anymore. Okay, so that's your competitive inhibition scenario. So what's happening with the other one? Allosteric or regulatory site case. So let's check out the diagram here. So if I have an enzyme, it can have multiple binding sites. Okay, so it's an active binding site is right here, the yellow one, and then the allosteric binding site is right here. Okay, so these inhibitor molecules or regulator molecules, I can have both types of regulators here. If these molecules come and attach here, it basically impacts the rate of substrate to enzyme binding. So the substrate is binding right here, right? So this is a different spot for the substrate. However, because of a new molecule coming in and sitting at this space, this impacts the rate of the substrate binding. Okay, it can be increased, it can be decreased depending on whether this allosteric site has got a regulator molecule or an inhibitor molecule. Based on that, this rate can be increased or decreased. Okay, conceptually, so now I can have the enzyme as idle, so nobody has bound to the enzyme. I can have an enzyme substrate complex, so only the substrate has bound to the enzyme. I can have the enzyme inhibitor complex, which means only the inhibitor has bound here. Or I can have the enzyme inhibitor substrate complex, where both the inhibitor and the substrate has bound, because these are 2 different sites on the system. Okay, so if you have a system like this, so I have 4 states in the system. I have the enzyme by itself, enzyme substrate complex, enzyme inhibitor substrate complex, and just the enzyme inhibitor complex. I can have 4 states. I can have state transitions, forward, backward, K1S, So this is forward K1 times S, backward K minus 1, so on and so forth. You have all these rate constants that you define and all. Only this state, the ES state, forms my product, right? So only when I go to the enzyme substrate state, which is right here, only then I can form the product. For any of these cases, if I am at EIS, if I am at EI, or even if I'm at E states, they don't form the product. So this becomes the most important state in this system with this inhibitory complex and all. Okay, so going back a couple of slides, so we were talking about this second scenario. That's what happens with allosteric or regulatory sites. And then the active sites are simple. This is simple competitive inhibition, as we saw over here, that an inhibitor has stuck to that site. The substrate cannot attach to it, so it is forming a new complex which cannot produce my products anymore. In this system, I can now write the differential equations very easily because I have two reactions over here. I have SE, C1, EP, and C2, all of these. So I write down the differential equations for all of those. And then at steady state equilibrium analysis, you already saw that we can consider dC1 dt, dC2 dt, the complexes getting formed, that is equal to 0. So no new complexes. The complex concentration is not changing at equilibrium. Once you consider that equilibrium correction over here, I can now solve for dPdt. How much is the product for me? So dPdt is my V, which is the rate of reactions at the end, right? So how is the rate of reactions of the product changing at the end? We come up with this expression right here. Now correspond this expression right here with what we saw in the normal enzyme kinetics case. So I'll just go back a few slides, and when I say normal enzyme kinetics, I'm talking about this expression right here. Vmax S divided by Ks plus S. That's the normal case, no inhibitors. Okay. When I did have the competitive inhibitor, Vmax S divided by S plus, consider this as Ks that we saw from the previous slide, but I have this new expression right here. So that's the addition that has happened because of the competitive inhibitor, which means that this term is greater than 1 because I have 1 plus something, right? So this term is greater than 1. I have 1 plus, in the previous case, I only had Vmax times S divided by S plus Kn or Ks, let's say, which is Ks is the rate constant. In this scenario, Km is the rate constant, but 1 plus something. What does it mean when I compare these two expressions? It means that the denominator is larger in this case. Denominator is larger in this case means V is smaller in this case, right? If my denominator is larger, I'm producing less P, or basically the rate of reactions, the number of reactions getting formed is V, which has gone down because of this additional term in the denominator. And that's what inhibition does. Inhibition is supposed to bring down your rate of reactions, right, from the enzyme kinetic system. So that's why solving these problems mathematically and doing a simulation of this and all that, these basically are not just qualitative, this is also telling you by how much my reaction rate will go down when I have this type of competitive inhibition. So these types of systems have been very well studied and have been quantified and validated and all of that. Right? So we are just doing a brief mock-up of this. So this is a little bit more complicated system because I have 4 different states involved over here, the allosteric inhibition and all, but you follow the same strategy. You basically write down the differential equations, you equate them to zeros for equilibrium concentration, you do some algebra, and you come up with something very similar. Again, you have this extra term right here, just like the previous case, which is telling you that with this inhibition, with allosteric inhibition as well, my V has gone down. So V has gone down because without any inhibition, I wouldn't have this extra term, but with the inhibition, I have this second term, right? So that means my V has gone down over here. Okay, so similarly, you can do more advanced methods right now. So cooperative inhibition, enzyme can bind more than one substrate molecule at the same time. So I have an enzyme, I have multiple sites, binding sites on the enzyme, and it is forming C1, which is C1 basically is a complex with one substrate, and then S can still bind to C1 to form C2, which is the same complex with two substrates attached, right? So this is my C1 with one substrate attached, and this is my C2 with two substrates attached. Okay, so you can actually now make it more complicated, that okay, if my enzyme is a large macromolecule, it has got 10 different binding sites, I may have C1, C2, C3 all the way up to C10, because it can form complexes with one substrate, two substrate molecules, three substrate molecules, all the way up to 10 substrate molecules, right? So, and then the same system, it is exactly following the same setup. You write down your differential equations, you do your equilibrium analysis, and you basically figure out the V. So in this case, with two substrate molecules, you will see a quadratic term coming up. In the previous cases, I was only working with single S. So my product formation was linearly dependent on the substrate concentration, S. Okay. In this case, when I consider two substrates can attach to my enzyme, my product formation is dependent on, see, this S and this S. You do a little bit of what you call the Hill equation. You will see, if you look up systems biology, Hill equation, you'll see it everywhere. So these are approximations to the system. So for this enzyme kinetics system, I'm making an approximation saying that, just hold on a second. I'm making an approximation saying that, okay, let's assume that K3 is infinity. So this is very fast. K3, this means C1 to C2 formation is very fast. Okay, so C1 is forming C2. This is happening very fast. Okay, K1 is 0, tends to 0. So which means that from E to C1 formation is very slow. So if you make some of these assumptions, it allows you to simplify the expression. So this is the actual expression of the two substrate binding case. And if I made these expressions simpler, I come up with a much more simpler expression like this. The bottom line is, Hill equation says that based on these rate constants and making these assumptions of the rate constants, Hill equation says that I can have multiple binding sites on my enzyme molecule, and depending on the number of binding sites where a substrate can attach, it basically grows exponentially. So if I have two binding sites, it's S squared. My rate of reactions is dependent on S squared. If I have three binding sites, the rate of reaction depends on S cubed, so on and so forth. And you will see many systems biology simulations that people do considering n binding sites. And this has been validated, these types of models has been validated. If you say I have n binding sites, you can very easily write down how product is formed. You don't have to solve your differential equations and all of that anymore. People have done it already for you. Okay, you can simply use this final form. So if you see a system of these types of models where people have simulated the system, and they say that we assume that this reaction follows Hill equation, which basically means that when you're working with larger protein complexes, they have multiple binding sites. Okay, and Hill equation is used to approximate the rate of reaction in such cases very well. Okay, so whenever I have large protein molecules doing a reaction, it will have multiple binding sites over there, and depending on the number of binding sites, based on the protein structure, you know what is the number of binding sites. You can use the exponential formulation of Hill equation to approximate that reaction, right? So it's just S raised to n, where S is the number of substrate molecules that binds to that reaction. Okay, this gives rise to the Hill's plot and so on and so forth. It's a very popular technique over here. Okay, long story short, plot of the substrate concentration. What we have learned here is, as I keep increasing the substrate concentration, my reaction rate, which is my dP/dt, right, at the end, it is no cooperativity is right here. This is the normal case. This is the normal enzyme kinetics case. If I have positive cooperativity, which means that it is making the reactions faster, then dP/dt increases right here. That is the highest value. If I have negative cooperativity with the inhibitors, my dP/dt falls much below. Okay, so this is the kind of, and basically with concentration you can find the equilibrium values of all of this. once you have trained your models and got your rate constants, you can also look at the transient values. We call it, before it reaches equilibrium, how did its concentration change in between? So I can get to that level of details too. Okay. Next part here is, this becomes even more useful because now you have ODEs, differential equations. You can numerically integrate these differential equations. I won't go into these Euler approximation and all of that. I will just give you the feel of it, is that whenever you have written differential equations and you have figured out your rate constants, right? What are the values of the rate constant? You've fitted it with experimental data and found your rate constants. You can actually get expressions for S. So I can now plot how S is changing. I can plot how E is changing, C, P. I can analytically solve this as well. Okay, so I can just write down an expression for how S is going to behave after a certain point of time and all. Okay, it gives you full power of being able to play with this dynamical system. And that's where these differential equation based systems are very useful. So let's move forward. One point to check whenever you're working with differential equation based systems. So after this mini course, let's say if you are interested in doing your first kinetic model with differential equations, one of the things you have to mention there is what is the delta t, which means that how much you want to increase your like the time period, right? After this much time, I want to recalculate the concentrations and all. If your delta t is too large, your accuracy will fall. If your delta t is too small, it takes longer to solve these equations. It takes longer to simulate. So that's the trade-off. So that is a constant factor that you will have to initialize your system to. You'll see that the actual solution is in red, and this black line right here is for delta t very small. It is matching the concentration of what it should be, the actual red line. But if your delta t is very large, you can see behavior like this. So if you're not using that delta t, which is an initialization of the system, so you'll have to initialize your delta t to something. People generally keep it to less than microseconds to nanoseconds. That's what they keep the delta t to. Only when you are keeping it to such a small value, that's when you can get a good approximation of the kinetics of the system. If you're using milliseconds range or seconds range for your delta t, it is going to fluctuate a lot. Okay, so let's move forward. So we have about 30 minutes left. So I'm going to move very quickly. So stochastic models, it is actually a little bit harder than what we have just learned for the differential equation based system. So I'll keep it a little bit more abstract. Remember, the first point here was, what was differential equation based system assuming? That if I have A plus B forming C, so this is, let's say, A, this is B, and this is C. Let's assume that. So I'm going to use a different color. So let's say this is my A, B, and this is C, right? A plus B forming C. Differential equation based system is saying that, okay, if this is the rate K1 of this reaction, then given so many molecules of X, so many molecules of D, I will always produce so many molecules of X dot Dn, right? So many molecules of C. There is no, what do you call, probability involved. I will always produce this end product at this rate, right? So that's what that's the assumption that you make. So we basically call it a deterministic system. So if I'm simulating this system multiple times, it will produce the same result because there is no ambiguity here. There is no probability involved here. But an actual system, so this is what you write, your differential equation over here, and of course it does not have a probability. When it does not have a probability, it will always be deterministic. It will always produce the same results all the time. However, people have shown from the year 2000, it's been 20 years, a lot of work has gone in to stochastic simulations of biological systems. And everybody has shown that biological systems have a lot of noise over there. Okay, so the current view of protein production, if you're trying to plot experimentally how much protein is being formed with time, you're going to see that they get formed in short bursts at random time intervals. Okay, short bursts. So basically, your protein result, let's say a new protein is getting formed, you're not going to see a curve like this. This is a continuous curve. You're not going to see something like this. Instead, you will see nothing's happening, a burst right here, nothing's happening, a larger burst right here, nothing's happening, a smaller burst right here. Okay, That's what an actual system would look like. But differential equation-based systems gives you a nice little, continuous plot like this, which generally doesn't happen in a system. And people have used Schrodinger's law to express this type of randomness. They basically say that the amount of randomness in the system is proportional to 1 over square root n, where n is the number of particles in the system. Now, what does it mean? Like in very simple terms, it means that if my n is going down, if I have too few molecules, my randomness is growing up. Okay? If n is very small, you're dividing by a small quantity, so the overall value increases, so your randomness increases, right? At the same time, if n is very large, if n is high, the randomness falls. Now, what does it mean in terms of system simulation? So now, if you want to simulate a system, the first call you have to take is, does this system have a lot of molecules, or does it have very few molecules? Okay, so what kind of system am I looking at? One of the systems that I will show as an example at the end has got a large number of molecules in protein aggregation and all. Okay, so where we do in vitro experiments and all. But most in vivo scenarios, E. coli cells and all of that, the number of protein molecules over there is very little, less than about 10,000 copy numbers of some of the proteins. So if you have very few molecules, it will show a lot of stochasticity, and your differential equation based model will fall apart. It is not going to give you the right kind of dynamics. You must use stochastic simulations if your number of molecules is very low in the system. On the other end of this is if your number of molecules is very large in a system, so let's say the protein aggregation system that we will talk about later on, if your number of molecules is very large over there, you can actually just use differential equation based systems in scenarios like this. Okay. So noise in gene expression, there is something called intrinsic noise and there is something called extrinsic noise. Intrinsic noise happens because of the low concentration of molecules. And if you think about it, if you think of it from a physics perspective, what does it mean? My reaction A plus B forming C, what's actually happening is a molecule of A is coming in contact with a molecule of B. So they are colliding. Only when they collide, that's when they form a complex, right? So if I have very few molecules of A and B, my chances of collision is very small because they are diffusing in the system, right? So if I have 5 molecules of A, 2 molecules of B, the chances of them coming together is very small. If I have 100 molecules of A and 100 molecules of B in the same volume, the chances of them coming together is much larger. Okay, so that's where the randomness comes in. If you have fewer molecules, you have to go for probabilities and stochastic simulation. If you have more molecules, you can actually work with differential equations, which can still produce good results for you. Okay, so that's the difference between these two simulation paradigms and all of that. So that's for the low concentration component. Now, if you're looking at the extrinsic noise, that is because of other factors. Number of ribosomes. Yeah, Victor, you have a question? Go ahead.
Yes, what constitutes a small concentration? What sort of system?
So inside the cell, if you're looking at simulations inside, that's a good question. So if you're looking at simulations inside the cell, they are always much small copy numbers. So numbers in the range of thousands, okay? 1,000 molecules, 10,000 molecules, that's considered small. You must do stochastic simulation. One of the examples that I'm going to show is with A-beta aggregation systems. It's a protein aggregation system. That's where we are working with micromolar quantities. If you have micromolar, multiply with Avogadro's number. So the actual number of molecules is 10 raised to 10. That's a much larger value. Over there, differential equations work fine. So if you're working in the molar range, if you can convert it to moles, and you are working at micromolar range and so you're okay. You work with differential expressions. If you're looking inside the cell, in vivo system, where you are working with thousands of numbers, so 10,000 number and all of that in the number of molecules, their differential equations will not work. You have to do stochastic simulation. Okay, and some of the packages, I will talk about Copper C in a little bit to show you an example of how these are simulated. So most of these packages have got both the stochastic simulator and the ODE simulator. So it's your choice which one to use. Once you have defined your set of reactions, you just don't care about equilibrium analysis and all that we just did. You just hit a button and ask it to simulate. It will do it for you, right? Of course, we did look into some of the math so that you understand what is happening right behind the black box. But once you have understood the set of reactions, you can simulate them. It's no big deal. And all of you can actually just do it for fun. Okay. So let's just move forward. So this is called the stochastic phenomena. There is something called stochastic resonance, that these systems, the probability values, they work together to make a few things happen. It's a dynamic nonlinear system. A lot of math goes in to try to simulate this. I'll just push forward. Give you an example here. So this is an enzyme catalysis system. These are some arbitrary values over here. So don't take it in the face value. So the difference happening is, if my initial concentration of E and S, enzyme and substrate, initial concentration is, let's say, 1000, I'm still seeing stochasticity. It's not a single line. You see, it is jittery, because multiple experiments at this time, multiple experiments is giving me different points for this curve. So that's why it is jittery. That's why you have this spread. Okay. But if I worked with 100 molecules, you see the amount of jitter is so much more for each of these curves. So basically, I've done multiple experiments with fewer molecules, and at this time, I fix the time. At this time, I see that this red curve, it is giving different values across that mean. Okay, so fewer molecules, more the jitter. So multiple experiments will produce different values for you. Okay, a differential expression-based system will only give you a single curve like this, a single curve. This does not help because my actual values are falling all over that curve. Okay, so I should not be able to, I cannot quite fit the differential expression-based single line to these multiple values that I'm seeing from these multiple experiments that has happened over there. Stochastic simulations gives you this kind of a spread. So for stochastic simulation, it doesn't give you a single line like this. It will give you these types of points all across this. Okay, so now you can actually do an R-squared proper comparison fitting with the experimental data, and that will give you a better feel of the rate constants and all of that. So that's why whenever you're working with smaller number of molecules, like an in vivo system, go for stochastic simulation. But if you want to just train your model, you do an in vitro experiment, you're working with larger concentration there, you get the rate constants and then run a stochastic simulation to predict what will happen inside the cell in an in vivo scenario, right? So that's what people do here. So when it comes to stochastic simulation, in the interest of time, I will just skip chemical mastery equation. So this is the math behind the stochastic simulation paradigm. So let's not think about that. There is one guy, Dan Gillespie, who solved the stochastic simulation system back in the 80s. Late 70s, 80s. Nobody took it seriously back then, but now everybody realizes that his stochastic simulation algorithm is the way to go for these types of systems. Gillespie, so basically when you're doing a simulation, a stochastic simulation, you have to work with probabilities. So this is where, like he's looking at, remember we talked about molecules coming together. So the basic idea here is, I have molecule of A and I have molecule of B. They will react when they have come together. And you're looking at, so if you consider both of them as spheres, this is the radius of the two spheres. So let's say this has got radius of R1, this has got radius of R2. Okay, so now what Gillespie did was, he said that, okay, let's say that this is a larger radius of size R1 plus R2. So a reaction will happen when the two molecules have come together. If they are not coming together, no reaction can happen, right? So they have come together means that I have a larger sphere like this of total radius R1 plus R2. So now he's saying that if I were to compute the probability, I will let this larger sphere move in time to form a cylinder. And the volume of this cylinder is pi R12 squared. So R1 plus R2 is considered R12. So pi R12 squared multiplied by V12 times delta T. V12 is the relative velocity of R1 of V1 of molecule A1 and molecule 2. So basically this curves out a sphere in the system. that the two molecules are together and they're moving in the system. In order to compute the probability, he's now saying that, okay, this is the overall system, overall media. This is the total volume, capital V. Out of this total volume, capital V, the probability of this reaction that will happen is if you fall inside this cylinder. Because this cylinder is telling you A and B are together, and I will let this complex move around in this system. Right? So whatever cylinder it has formed, but this will not traverse the entire system. So that tells me the probability that this is my chance that the molecules have come together instead of like considering this entire space. And that's the idea that delta V, this is called the collision volume delta V divided by the total volume capital V. And you can come up with this expression pi r12 squared multiplied by V12 multiplied by delta T. Okay, so that's the basic idea behind it. This is called collision theory, where the main idea is two molecules can react only when they come together. And in order to do that, you're looking at the entire volume of the space and things like this. Okay, so let's move forward. And based on that, there are also other things. So I have C mu is the rate constant in the stochastic simulation, whereas K mu is the rate constant from the differential equation simulation. Remember, I said that, okay, you work on an in vitro system, you find out your rate constant K mu, but then you can do a stochastic simulation for the in vivo case, a stochastic simulation for the in vivo case, and over there, you basically can map the rate constant you got from the differential equation system to the stochastic system, right? So what is the relationship between K mu and C mu? Considering the expectations. So this is where the probability distribution comes in. We're saying that the joint probability distribution of x1 and x2 is the same as separately multiplying the expectation of x1 and x2. Expectation means concentration. So at that time, the concentration of x1, it follows the distribution, because now we are talking probability. It's not a single value, right? So it's saying that the mean value at that time multiplied by the mean value of x2 at that time, they are independent. So if I were to look at the joint probability of the two, I can write this x1 times x2, the joint probability of x1, x2, the expectation of that as expectation of x1 multiplied by expectation of x2. If that's the case, the numerator and the denominator cancels out, and this is left as b times c. Okay, so again, long story short, for you guys to work with stochastic simulations, what you assume is, okay, this is the rate constant for the differential equation system. I have fitted it to my model and all of that. What is the rate constant that I should use for the stochastic system? So to get C mu, all you do is the rate constant from the differential equation system, K mu, divide by V, which is the volume of the compartment that you're using. That's it. That's all you do. You don't have to go into the math of it. So they have solved it for us, and we can just use that result directly. Okay, but that's the difference, because you cannot use the rate constant of a differential equation system directly into the rate constant of the stochastic simulation system, right? So there is a governing equation there. And then they have done the math behind it and came up with a proper simulation algorithm. It's a very nice, neat simulation algorithm. This is already coded in MATLAB. So if you're using MATLAB, you can do it. It is in Python. You can do stochastic simulation in Python. And Copus is a tool that is quite useful. I will show you what it does for the last 15 minutes. We are almost there. So I have one last topic to cover. So I'll do that in 5 minutes before we look into the demo. Okay, so this is about the simulation algorithm that is already coded in. You don't have to code this in. You are just writing your reactions and let the simulator do the hard work for you. Okay. The last topic in this mini course is Boolean network models. Okay, now this is also a very important topic, mostly used in genomics. Many of you are interested in computational genomics, and you'll see that Boolean network models are a very important mathematical paradigm over there. So why Boolean network? I'll skip the basic pieces. Okay, so think about it this way. Any genetic circuit can be written as a Boolean circuit. So if you have done computer science, you know what is a Boolean circuit, right? So Boolean circuit, so for example, this is an AND gate. So I'm saying that CDK, this is cell growth, CDK7 and cyclin H has to come together. Only when the form come together will it go to if this complex will actually work with cyclin E and P21 WAF1 in a NAND gate. So they are together and down regulated to form this product. So if this is the logic of gene regulatory system, it's up regulation, down regulation, right? So it's 1 or 0. Consider up regulation, down regulation as 1 and 0. So if I do this abstraction of 1 and 0, I can just write it in computer science language as a Boolean circuit, no problems altogether. Okay. Now, if you write it as a Boolean circuit, then I can use all computer science methods to figure out the dynamics of the system. Of course, what is the problem? We just learned kinetic models. It is much more detailed. I'm not saying 1 or 0. So it gives me a much better control of the system. What is the problem with Boolean circuits, Boolean models like these? It still gives me qualitative results. It does not give me the full details of the system because I'm just saying up regulation, down regulation, which is 1 and 0. I'm not using the actual value, right? However, if you do this, you can still understand the system very well. I mean, there are many pieces of it that we'll understand. So I'll skip some of these. Simple regulation, multigenic, pleiotropic regulation. So let's not worry about it. Let's say that this is my genetic circuit. Okay, these are my genes, and this is the network. So which means gene 1 is regulating gene 2 and gene 3, maybe plus sign or minus sign, up regulation or down regulation, right? So this is how the network is going to look like. Now, with this, with a Boolean model, I'll skip this. Let's not worry about synchronous, asynchronous, or wiring. Okay, let's check this one out. I'm just looking at the interesting pieces of it. Okay, so let's assume that I have 3 genes, A, B, and C. Okay, and these are the relationships. The edges are the relationships. So we're basically saying A upregulates. The blue line is upregulation. A upregulates B. B upregulates A and C. C, red line, downregulates A. Okay, so that's my network. I have a network of 3 genes, and I know the relationships. I know A is upregulating B, B is upregulating A and C is downregulating A. Okay, so now we just said that upregulation, downregulation, we will consider 1 or 0. So that means if my initial condition, let's say, is 1, 1, 0, what does it mean 1, 1, 0? So A is 1, so it can do some changes in the system. So it is upregulated. B is 1, it can also do some changes. C is 0, it cannot do any changes. Right? What is the next state of the system from 1, 1, 0? So A being 1, it will upregulate B. Okay, so B is already 1. In the next state, B stays as 1. Okay, so B does not change because it's a Boolean system, right? From 1, it does not go to 2. It stays as 1. So that was for B. What's happening to A? A gets upregulated by B. So B is 1, means A will also be at 1. Okay, and C is 0 means nobody is downregulating A. If both of these, if C was also 1 and B was also 1, it would have come to 0 because this would have added +1, this would have taken away -1, right? But because C is 0, the -1 is not happening, so A stays at 1. Right? What's happening to C? C is right here. Since B is 1, it is making C 1. So my next state is 1, 1, 1. If you have 1, 1, 1, what happens to the next state? You see, A goes to 0. Why? Because B has pushed it up. So I got a +1 for B, and then I got a -1 for C. Right? So ultimately, my A got to 0. What's happening to B here? B is still a +1 because A is +1. It's keeping me to +1. And then what's happening to C? C is still a +1 because B is a +1 right now. It is keeping me to +1. The C is +1. Make sense? Okay. So once you know which gene is regulating which other gene, up or down regulation, then I can exactly say the state, how the state of the system is looking like, right? Just based on zeros and ones. Now, look, think about the complexity here. Why did we consider the Boolean system here? Because the state space is 2 raised to n. N is the number of genes that we are talking about, right? So if I have 1,000 genes, or let's say 10,000 genes, each gene can be either in state 0 or 1. These are its two Boolean states. So then if I were to write this truth table, how many different states am I looking at? 2 raised to 10,000. That's the number of states that I can have in the system. So that's why even if you consider Boolean approximation, it's still good, because you're still looking at a state space of 2 raised to 10,000, or however many genes you have. Boolean network algorithms, there are a lot of very cool algorithms that have been proposed in this Boolean network formalism. It gives you very interesting dynamics of the system. So for example, this system, we came to know that there is something called attractors, which means that if you have started from this state, once I've gone to the 0, 0, 0 state, it cannot come out of it. It will keep cycling right here. It will keep cycling in this state 5. It can never come out if I started from 1, 1, 0. However, it depends on the initial state. If I started from 1, 0, 0 instead of 1, 1, 0 in the previous case, same system, if I started from 1, 0, 0, so basically I'm perturbing the genes. I upregulated somebody, I downregulated somebody, and all that. Right? I gave it a different initial state. If I do that, this system, look at this, it is forming an attractor cycle. So basically, once you go to state 2, from 2 you go to 3, from 3 you go back to 2. So it is cycling between two states infinitely. It cannot come out of it. Does it make sense? So this is my state 2, and this is my state 3. So the system is toggling infinitely between states 2 and state 3. It cannot come out of it. But these are very strong results where you're basically saying that, okay, you give me a gene regulatory system, I will initialize it. I have done the math with the Boolean networks, I've done the simulation, figured out the attractor states. I will tell you, perturb the genes this way, upregulate this guy, downregulate this guy, so on and so forth. Create this Boolean map for me. And then your system will result in this final end state. You're basically telling them, the experimentalist a priori, what kind of end state may I reach with the current state of the gene regulatory network. The challenge here is to know the exact network connections. For E. coli and yeast, we already know who is regulating whom. A is regulating B. Is it an upregulation? Is it a downregulation? We already know that, right? But for human, for mouse, or for many others, the regulatory network is not known well, and that's where some of the challenges come in. Okay, so people have done more on attractor dynamics, and these are results from yeast and E. coli, and they have actually demonstrated these different types of attractors that have been seen in these systems. So attractors is a very important component of dynamical systems. I'll just move forward with this. One of the main problems that we have is to figure out which gene is connected to which other gene, right? Is it a upregulation? Is it a downregulation? So on and so forth, right? That's one of the most important pieces we talked about. So we have many algorithms that have been proposed, and you can actually look up the reverse engineering area, reverse engineering, or ADN knows it. So you can actually talk to ADN about it as well, or inference algorithms. So network inference algorithms. So I will just say network inference. So what this is trying to do is this is trying to predict which gene is connected to which other gene. Is it an upregulation? Is it a downregulation? And all that. Once you know this wiring, you can do the Boolean network formalism. You don't have to go into the actual kinetic modeling that we saw before. Kinetic modeling is more detailed, but Boolean network is in the middle space. It is not that detailed, but it can tell you what is the end state that you're looking at, right? What kind of different end states can I go to? Perturb the system this way, change these genes to 1s and 0s, and then you should see a very different end state for that system. Right? So it's also a very cool way of controlling your gene regulatory network. Some methods that people have used here: information theory, Bayesian networks, machine learning, deep learning, decision trees, differential equation-based systems have also been used to infer these types of regulatory networks. So we are very badly, out of time. So I will just skip over to one very quick demo. So we have been talking about kinetics mostly in this system. So for this demo, we are looking at Alzheimer's disease, and there is something called a protein aggregation problem that happens here. So basic point here is there are some monomers in our brain, A beta monomers. They react to form dimers, from their trimers, tetramers, all the way to large plaques in the brain. Okay, people initially thought that these large plaques are the ones that create the toxicity for Alzheimer's disease, but now people say that no, there are some other molecules that arrest the growth. The smaller oligomers, trimers, tetramers, and all of that, the smaller oligomers are more toxic to the brain. In order to understand the toxicity of Alzheimer's disease, you have to be able to simulate this system. Now, this system is pretty cool because it's a very simple system. I mean, I don't have any other types of molecules to consider, and mostly the measurements are from in vitro tests and all. But it is also quite complex because think about it, from monomers, so let's say from A1s, I can go all the way up to A5000 mers, because that's how long my fibrils are, these plaques are in the system. Okay, so I can actually go to that level. So what does it mean from A1 to A5000? Think of chemical kinetics. Think of the kinetic model. I'm going to write differential equations dA1 dt, dA2 dt, all the way up to dA5000 dt. So now you have a system with 5,000 differential equations. And that's what makes it fun, because that's when you need a computer science person to come in and say that, how do I simulate this system? What kind of approximations we do and things like that, right? So when you're looking at systems this complicated, where you have so many differential equations and all, we cannot use off-the-shelf types of simulations and all of that. That's when we go into some of the, tools that are available. So one of the tools that I will strongly recommend you guys looking at is called Copasi. So you can see my screen here, and I will just go to the about. So Copasi, you can see Copasi 4.31 build 243, all of you, right? Over here. So Copasi is a tool for these types of simulations. You can do stochastic simulation, you can do ODE-based, differential equation-based simulation and all. Just type up Copasi, it is free to use, okay? Any and every one of you should be able to download Copasi on your desktops, and you can now run simulations for any system. For the Alzheimer's disease simulation, it is actually much more detailed than what we have covered here. So for example, over here on the left, you see compartments. So for this system, I said only a single compartment, one compartment, keep it to one compartment. You can add more compartments to your set. So you say that these are the reactions in the cytoplasm, these are the reactions in the nucleus and stuff like that, right? You can create different compartments. You can have molecules coming from one compartment to the other and all. So you can build much more detailed simulation systems like this. Species, let's say monomers, dimers, trimers. I have only gone up to A11, A12. So let's say this F is getting formed at the very end. I'm only showing this as a demonstration for 12 species. So A1 plus A1 forms A2. So if you look at the reactions, what types of reactions we have? A1 plus A1 forms A2, A2 plus A1 forms A3, A3 plus A1 forms A4, all the way up to A11, A11 plus A1 forms F. This is a modeling abstraction. So if you don't want to write so many reactions, you just say that A12 is enough. I don't want to write other differential equations beyond A12. So I've just approximated A11 plus AF plus A1 to make an F, which is, let's say, a fibril. And then from there, after I have created fibrils, the fibrils can take up all the other oligomers, A1, A2. So F plus A1 still keeps it an F. So basically, I'm saying that it is still a fibril, but it's not growing as much. So this is a modeling abstraction. You can write all 5,000 differential equations as well. You can write all 5,000 different reactions as well over here. Once you've written these reactions, it automatically creates your differential equations at the background. You don't have to write the differential equations, right? So that's the fun part of Copasi. You just write down your reactions this way. It will create your differential equations. Then this parameter estimation becomes important. So you see this parameter estimation box over here. You can set up the experimental data. So I have a CSV file where I have the experimental values, right, of one of the species that I want to fit it to. I want to know the rate constants. So in order to do that, I need to fit it to experimental values. Those experimental values are stored in the CSV file. Okay. So based on, I have got the experimental values of F. Remember the F species we saw? So we have the experimental measurements. With time, what is the value of F? What is the concentration of F with time? Okay, so once you have those experimental values like this, you set it up on the experimental data set and you just hit run right here. Okay, what it will do is the parameter estimation block, what it does is it creates plots like this for you automatically. So these dotted curves are the experimental values, right, with time. So the x-axis is time. So after so many hours, so let's say after 1 hour, 2 hours, 3 hours, so on and so forth, these are the experimental values with time. These are the dots. And then this trend line that you see is what the simulation is fitting it to. So I have a simulation of A1 plus A1 forming A2, forming A3, A4, A5, so on and so forth. It is telling me what are the values of the rate constants that will fit your simulation line, which is this, best to these experimental values. So best means what is the highest R squared that I can get. So it is doing an optimization algorithm at the background, right? This is actually a hard problem. It is trying to run some random search algorithms. It is trying to say that, okay, rate constant K1, let's put this value, run it. What is the R squared? Rate constant K2, let's put this value. So it's a random search that is going at the background. It's a very difficult problem to solve, but then they have perfected it. There are some very good algorithms for this parameter estimation toolbox, and you don't have to worry about it because Scopus is already has got them implemented. You just run parameter estimation. You can choose which algorithm to use, but most of them work well enough. And it will tell you what are the values of the rate constants that were achieved. So I believe the rate constants should be somewhere here. Oh, I'll have to check out. Okay, so for the parameter estimation. Okay, you can actually do sensitivity analysis of this in one of these places. Okay, you should have the output specification somewhere here. Plots, report templates. Yeah, so I mean, at one of these places, you can define the units of your rate constants, the units at which you want to simulate this. Is it a molar level or molecules level and all of that? You can put in noise. You can bring in some approximations to be done based on noise. You can do parameter scan. You can also do parameter identifiability analysis. Yeah, Victor, I know you have.
And perturbations?
Perturbations?
Yes.
So you see something called events right here. You see that I don't have an event added. You see something called events, 0, right here. Okay. What this does is, so I'm saying that, okay, I am running this system with initial concentrations of A1, A2, A3 and all that, right? But let's say after 5 minutes, I want to include a new species over here, or I want to throw in more molecules of A1. These are called events. These are called perturbations to the system. So you can actually add those types of cool things as well. You can say that, okay, I want to control this system, but not initial condition. I don't want to just dump in molecules or concentrations of the initially. I will let it run for 1 hour. After 1 hour, I will bring in so much concentration of species 5, 6, and 7. You can actually code it in through the events tab over here because that's a new event that you have added. And it will simulate just fine. It will do this rate constant estimation, the parameter estimation and everything for you at the background and give you what are the right values of the rate constants to match these types of experiments that you use. It's a very good tool, the Copasi tool. I will strongly recommend all you guys to just play with it because all you're doing is button clicks here and you're just writing the reactions that you want to simulate, right? A very rich tool. They have parameter estimation, parameter scan. So basically they can also do parameter scans, which tells you, is this parameter truly identifiable or not? So if I have only one experimental data and 5 rate constants to estimate, I will always get some values doing some fitting, but that may not be good values, right? So basically it tells you that, hey, this is the confidence interval of this parameter. Even if I got a fit for you and this is the value for this rate constant, I don't have much confidence on this value. It can tell you that. It can tell you the optimal number of experiments required. It can tell you that, okay, is this your model system? So many reactions. I want 5 different experimental plots. Don't just give me 2 because I cannot give you good confidence intervals with those 2. Give me 5 different experiments, run the experiment 5 times with different initial values. and give me 5 different plots, then I can give you a better confidence interval. Those are the parameter scan and parameter optimization toolboxes here. So very good tool. So if you are wanting to get into modeling and simulation of biological systems, please give this tool a try. It's very useful moving forward. Okay, so with that, I will stop, and I know we are over time, but we can take some questions from you all.
Si tienen alguna pregunta para el profesor Britton, tanto los que están acá en Zoom como los que están en YouTube. Victor.
Yeah, again, I'm sorry.
No
I have
problem.
a monopoly here. So what about if I want to start studying systems biology, let's say by myself for the moment, what literature should I
There's a big literature. You saw, Victor, so we covered a few different things here. At the very beginning, we looked at statistical tests, right? Clustering algorithms and all of that. That's a field in itself, okay? Then we got into kinetic models, differential equations. That's a field in itself. Then we got into stochastic simulations. That's a field in itself. And finally to Boolean networks for understanding attractors of gene regulatory networks and also reverse engineering. That is also a field in itself. The point is, you don't have to go into the mathematical descriptions of all of this. So what we covered in this mini course over here, this is good enough to get an introduction of what each of these pieces do, okay? But there are readily available tools involved that is out there already, okay? So Boolean network formalisms and regulatory network techniques, there are some web tools involved, and they have the tutorials for that. Okay, so go over their tutorials. Okay, and you can just contact me anytime, send me an email, and I can recommend some tools to you. Contact ADN as well. So ADN has worked with computational genomics a lot. So these statistics tools, reverse engineering, maybe even Boolean networks, he can help you with some of those tools. Boolean networks, the probabilistic Boolean networks package I know is available as a MATLAB code already. So you can just download that code, and you should be able to do probabilistic Boolean networks and figuring out attractors of your system and all that. Differential equations-based systems use COPACY. Stochastic simulations use COPACY, right? They have tutorials already. So if you go into COPACY, they have some example systems and they have tutorials. And that's where you start. That's where you get started. Okay, this is how I want to simulate your system, because you want to be a user of the system to be able to analyze your system of interest, right? You want to be able to simulate it, fit it to the experimental data and all that. You're not trying to develop any components of the system. Now, if you want to develop some components in the system, say that, okay, I want to create a better parameter estimation toolbox, which is essentially an optimization problem, because this is the experimental data, and how do I change the rate constants? There is gradient descent algorithms that people use and all of that for this. How do I change them so that my system can match the experimental data that I'm seeing? Okay, so those are hardcore computer science related problems that you are getting at, right? But to start with, you should start with each of these tools. So COPACY is one. Try vCell. So V is for virtual and cell. vCell. vCell is another web-based interface, and that's also a tool that you can freely download that also does system level simulation. They have the better support for stochastic simulations than this one, than COPACY. COPACY is much better for differential equations and all. Okay, so keep yourself to vCell and COPACY, and just check out their tutorials and examples. That will be enough for you to get started working with these systems, right? Now, genomics, if you want to get into genomics, that's a very different area, and there are other tools that is out there, statistical analysis tools and Boolean networks models and whatnot, clustering algorithms and so on and so forth. Right? So that's a very different area that will require some different kinds of things. But again, like one of the best ways of looking for it is talk to ADN. He knows about the genomics side of things quite well, and he can give you some links over there. And you can talk to me about the modeling simulation tools like vCell and COPACY and things like this. Okay, that's where you get started. Of course, when you're hitting some bottlenecks and all of that, that's when you may need some better expertise, somebody to help you out with those types of systems. But for most simple systems, you should be able to just play with these tools. Just read up their manuals. It will tell you what each of these boxes are doing. And that's all you need to know. Okay.
Thank you.
Thanks.
Thank you so much, Dr. Gus.
Thank you.
For me,
Good
I
to see you
appreciate your
again.
presence here.
Yeah.
See you tomorrow in the main conference
at 9 o'clock
PM.
US time, right?
Yeah,
Yeah. Okay. Okay.
Thank you
And
so much.
that's 1 hour, right, Adrian?
Yeah, only 1 hour.
1 hour with questions and everything, because I can then.
No, only it's 15 minutes and 10 minutes for a question.
So it's 1 hour, 10 minutes. Oh, 50
Yeah.
minutes plus 10 minutes.
Yeah, it's 1 hour, 15 minutes, and 10 minutes for a question.
Okay. Sounds good.
See you tomorrow.
See you. Thanks all, everybody. Yeah. Bye.
Estamos volviendo después de las 2 de la tarde con las siguientes ponencias.